the sponsorship dari Badan Pengelola Dana Perkebunan Kelapa Sawit BPD PKS, Provivi Incorporation, PT Bayer Indonesia, PT Syngenta Indonesia, Corteva Agriscience, Crop Life Indonesia, PT Sanjoy Agro Indonesia, PT Tangguh Agro Unggul, Alister Indonesia, Crop Care Indonesia, PT Astra Agro Lestari, PT Agrikon Indonesia, Asosiasi Produsen Pestisida Indonesia, Peladang Agroscience, PT BC International Tbk, PT Petrosida Gresik, PT Inti Everspring Indonesia. Thank you for your supporting us in this international conference. Ladies and gentlemen, before we start the conference, we would like to inform that the semi-final of Linear Games 1 will be start. For the participants, please be prepared and go to room A with Dr. Rina Rahmawati as your license officer leader. Good luck and see you for the semi-finalists. And ladies and gentlemen, let's move to the next agenda. That is the plenary one with our invited speakers. To leading this session, please welcome the chairman of Entomological Society of West Sumatra, Dr. Henny Herwina as our moderator. Time is yours.
Okay, thank you very much, our Master of Ceremony, Mr. Nando. Good afternoon, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. With the highly respect to Bapak Profesor Dadang, our President of PAE, the Entomological Association of Indonesia, and our invited speakers. For today, we will have three offline invited speakers, and then in the next session, we will have around five speakers that will be joined with us for the presentations, for the research by online. Okay, ladies and gentlemen, my name is Henny Herwina from Biology Department, Faculty of Mathematics and Natural Sciences of Universitas Andalas. I'm very happy to be here with you and to be a chairman of this session for the invited speakers of three of researchers from several areas worldwide. And I would like to invite our invited speakers, firstly, to Ibu Nurul Wahida Othman, Miss Nurul Wahida Othman from Universiti Kebangsaan Malaysia, to have a place in the stage, please. Please welcome our first speakers. And the next is Professor Dr. Nurul Huda from Universiti Malaysia, Sabah. Thank you. And the next speaker is Mr. Maulana Marman, SP from Provivi Inch. Yeah, thank you, sir. Please have a seat. Yeah, very nice to have three of our speakers, invited speaker now in this afternoon. And how are you, Ibu Wahida?
Yeah, I'm fine, thank you.
Okay, how about you, Bapak? We have a microphone here, and maybe before we start the presentation, I would like to know what's your impression about our activity from this morning until this time. Yeah, Prof. Wahida first, maybe. We have a opening speech with our governor.
I really enjoyed the opening ceremony.
Yeah,
especially the tarian piring, but it scares me a little bit when they step on the piring. But I think
tari piring.
yeah, it's well organized conference, I think. with the participant and the exhibition also outside. I'm looking forward for more presentation.
Oh, nice. Thank you so much. How about you, Professor Nurul Huda?
Okay, thank you, Ibu. I think it's a good opportunity to attend this global conference, maybe to learn each other from the speaker and also from the participant. Okay, thank you.
Okay, thank you so much. And next to Bapak Maulana, maybe can I call you like that?
Thanks, Bu. It's
Yeah.
honorable for me. The first time visit to Padang.
Mm.
Basically, I'm Indonesian, but never come to Padang. But it's the first time here.
Oh.
Very appreciate. Like Bu Wahida said that the tari piring is very enthusiastic. Then I worry blooding there.
Yeah, that's very unique art
It's
of
the first
us.
time for me to present as part of the entomological.
Yes.
Basically, I'm a special for the with size management. It's the first time for me to present as entomology.
Okay. Thank you very much for the impression about our program from morning until now. And we are looking forward the presentation from each of our keynote speaker, ladies and gentlemen, because every keynote speaker have a very interesting title and specific subject to share with us. And before starting, maybe based on our book program, we can start from Ibu Wahida and continue to Prof. Nurul and Pak Maulana. So before that, I'd like to ask some help from our operator to introduce some curriculum vitae of our speakers. Yeah, ladies and gentlemen, we have Ibu Nurul, Dr. Nurul Wahida Othman from Center for Insect Systematic Universiti Kebangsaan Malaysia. Yeah, and has you can see in the in front of us, the insect physiology and regulation is her research interest, also insect ultrastructure, proteomic, biopeptide development, and insect toxicology, mostly about insect physiology. Yeah, and the insect physiology and insect histology is her skill. And then the education is from Universiti Kebangsaan Malaysia UKM 2003, the magister program from UKM also, and doctoral from Australian National University, Canberra, Australia. Very nice. Okay, so Ibu Wahida, we'd like to give space and time for you for your presentation. Yeah. The floor is yours. Yeah, maybe you... Oh yeah, thank you. From Prof. Thank you, Prof. The title is The Potential of Wolbachia, yeah? Maybe Dr. Wahida will start herself. Yes, please.
Is it working? So, Assalamualaikum and very good afternoon to all of you. My name is Nurul Wahida. I'm from Center for Insect Systematic, as been introduced by Ibu Henny. So today, I would like to share a bit of my research on the potential of Wolbachia synthetic peptide, WOFA, for future biopesticide development. So we know that biological control is a pest management in agriculture and forests that utilize living organisms to control the population of insect pests. So biocontrol agent can be either from predators, parasites, or even pathogens like bacteria, and they attack the target pest species as natural enemies. But what if usually this biocontrol agent we use as a whole organism, but what if we only use a tiny parts of these organisms as a biocontrol tools? So everyone is quite aware about Wolbachia, but I want to talk about Wolbachia synthetic peptide, the WOFA. We know that Wolbachia is endosymbiont. This is intracellular bacterium, okay, that infects up to 70% of arthropods, okay, including insects, mites, crustaceans, and nematodes. And we know the infection of Wolbachia into the host, it can change the phenotype of the host. that is the cytoplasmic compatibility, the male killing, feminization, and also parthenogenesis. We have extracted a novel Wolbachia synthetic peptide, a WOFA, which customized from these conserved regions of the Wolbachia surface protein. Okay, surface protein in these bacteria is important as they interact with their environment. And this WOFA was isolated from wasps. Okay, this is Copius ericinus species. So it is economically important endoparasitoid species. So how it all started, why we use this WOFA? Previously, when we have conserved this peptide, we have tested on crickets, and we can see some changes in the immunology, especially the prostaglandin levels, and we can see formation of nodules. So I did a few studies on red palm weevil and invasive species. Okay, we know that this is a Rhynchophorus ferruginous. Okay, and this is invasive species for Palmaceae plants, especially in Malaysia. It attacks a lot of coconut tree. And you can see the symptoms like dropping leaves, cracks, damage on the petals. And the problem with this pests, you know the infestation, the lead infestation, because it lives inside the tree trunk and it's very hard to detect. Currently, I think there is still no effective control strategy to control these invasive species. In Malaysia, we currently use chemical pesticide like cypermethrin and also using pheromone trap. But the problem with pheromone traps is only target certain radius and it's not really effective during rainy seasons, okay? And the early detection is very difficult due to latent symptoms of the infestation by this pest. Talking about insect immune system, my study, we would like to see the effect of this Wolbachia synthetic peptide towards the immune system of this red palm weevil. Okay. A bit background about the insect immunity. In insect, the most important cells involved in insect immunity is the hemocyte cell. You can find various morphology of hemocyte cell inside this insect, such as prohemocyte. So this is similar to the stem cells of the hemocyte. So it can become plasmocyte or granulocyte or other types of hemocytes inside the insect. So these are the main components of cellular innate immune response in insect. So they develop very high innate response compared to acquired immunity system. Compared to vertebrate, we rely mostly on antibody, for example. But they are more to cellular response, means that these hemocytes will attack any pathogen that comes into their body. So questions to ponder. So if I want to study about red palm weevil and we would like to infect it with Wolbachia synthetic peptide, I need to know whether this RPW in Malaysia, today is already infected by Wolbachia. If it is not, what will happen to its reproductive system if we induce it with WOFA? And then what are the changes of the physiological immunity, especially the hemocytes, and also the changes in the protein hemolymph after the immune response with WOFA? So the objective of my study is actually to screen the presence of Wolbachia in RPW from different localities in Malaysia and to detect the changes in the hemolymph, especially is there any unique protein being expressed when we inject them with WOFA? and determine the hemocyte types, what is their immune reaction, and what are the profile of these hemocytes. And lastly, to see the changes morphologically or histologically towards the ovary of red palm weevil. So we did the screening, okay? From this study, this is the first record of Wolbachia status in red palm weevil in Malaysia. So we found that there is no Wolbachia detected from 60 samples of red palm weevil from 3 different localities. And this no infection of Wolbachia in RPW could relate it to the reproductive mode, environment condition, competition among symbiont, and high specialization of Wolbachia hosts. And then we look at the protein, expression of the protein inside the red palm weevil after we infect with Wolbachia. We found that 143 protein being expressed, and then we found 17 unique proteins. And this protein involved in the development, metamorphosis, and also reproductive, and they are downregulated, okay, during the treatment with WOFA. And 5 unique proteins, interestingly, were identified, detected from another endosymbiont in red palm weevil from this species, Candidatus nardonella. So means that the RPW immune system relies on this another endosymbiont to produce another unique proteins to protect them from the infection of this WOFA. So look at the hemocyte types. We can find 5 different morphology of hemocyte cells: the granulocyte, plasmocytes, and this is unicytoid, spherulocytes, and prohemocytes. So prohemocytes usually round shape, small, it's like a stem cell. The important hemocyte that we want to look at is actually this granulocyte and also plasmocyte. So it has spindle shape and it can change. This is cytoplasm. It can elongate if it want to attack pathogen inside an insect body. And this granulocyte contains a lot of granules. They contain enzymes also to attack the pathogen. So if you look at the RPW hemocyte types, the granulocyte and plasmatocytes, they have this pseudopodia present because this is the most active hemocyte cells that attack any pathogen that invade the RPW. And from the total hemocyte counts, this total hemocyte of RPW only affected by the concentration of the peptide. You can see the control, the increasing in hemocyte from the lower concentration, but higher concentration, the hemocyte reduce. Why? It's because these hemocytes being actively used during the immunity challenge. And hemocyte increase after treated with 0.83 millimole at 24 hours, but then it reduced. So hemocytes being produced and used, produced and used. So from this differential hemocyte count, DHC, you can see that the proportion of granulocyte, the hemocyte that has a lot of enzymes and granules, is the most major hemocytes found during the immune challenge. And then followed by these plasmatocytes and others. So after the immune challenge with WOFA, okay, at 24 hours after treatment, only granulocyte and prohemocyte percentage is increased. And the higher concentration treatment, all the hemocyte increase except for granulocyte. So 48 hours you can see that percentage of granulocyte increase, but lower than 24 hours. Okay, this is the control. This is the lower concentration, the purple one. The green one is the higher concentration. So control, after 48 hours of infection, it increased a little bit. And then for the higher concentration, it reduced. From the morphological changes, okay, we can see that there is an activation of immunity due to this. You can see changes in granulocyte cell. This is the control cell. So this is the one that has cytoplasm elongation. So it starts to activate, okay, to engulf and do the phagocytosis of a pathogen. And then this one, you can see a lot of vacuole, lompang. Okay, so it already released all the granules. And then this one also form a vacuole, and you can see cytolysis. You can see uneven border of the cell, and also lysis of the cell. So these morphological changes of plasmocyte, it also has this cytoplasm elongation. And hemocytes before treated with WOFA, you can see the control. Cells can be seen separately, but it started to aggregate. Means that they start to do nodules. Okay, they come together to attack the pathogen. So you can see cell aggregation. So changes in this cell morphology indicates that WOFA activates the RPW immunity, as we showed the cell abnormality, especially on granulocyte and plasmatocytes. So how about the formation of nodules? Formation of nodules is due to the melanization. So when something invades an insect body, the immunity system will trigger, start to melanize. When they aggregate, they will melanize, and they will kill the pathogen. Okay, this is after 24 hours. You can see black dots here. And after 48 hours, there are more black dots. It means that there are a lot of nodules, especially on the fat body. Okay, this is the RPW ovary morphology. So we would like to see, is there any changes of the morphology of red palm weevil, especially the ovary, after we do the test. So normally it has a pair of ovary and all these busa, coprolacic, the spermatica and vagina. It also has this lateral oviduct. So after treatment, there's a significant changes on the length of the ovary. And the ovarian index, after treated with WOFA, you can see that higher concentration of WOFA affect the morphology on RPW reproductive system. So it shows significant changes, means that the oocyte maturity in the ovary are affected. From the stain, okay, we use the H&E stain. You can see the control tissue, it takes up all the stain. But towards 24 hours, 48 hours, it's fade in color as it produces vacuole. All the granules being out, so you cannot see the stain clearly. And you can see different size of vacuole formation between the cells itself. So cells are not compact anymore. And RPW ovary histology, this is the normal cell. And you can see there's also changes on the nursing cell. So nursing cell, it gives nutrients, it takes care of the oocyte or ovum inside the ovary. And less nursing cells means that cell mitosis disrupts, and the development of this oocyte inside the ovary is actually affected by cytolysis.
Excuse me. Oh, okay.
Okay. So in
For the
summary,
summary.
so WOFA had activated the cellular immune response of adult in RPW, and it has potential to be developed as a biopesticide to control insect pests. So information about this prevalence and effects of peptides from endosymbiotic bacteria in pests may improve and optimize biological control efforts in future. With that, these are my team members that involved in this study. Thank you so much.
Okay, thank you very much, Dr. Wahida. And please have a moment because we will continue to the next speakers. Professor Dr. Nurul Huda. Ladies and gentlemen, we will have discussion session after 3 of the speaker will present their research to us. And next, we will invite Associate Professor Dr. Nurul Huda from Faculty of Sustainable Agriculture, Universiti Malaysia, Sabah. And the research interest of our speakers now is about food science and technology. And the experience skill of this professor is about food processing and preservation, food nutrition, and also halal food as well. And the education is from Universitas Bung Hatta, angkatan 1990. Beda 1 tahun kita, Pak. Oke, jadi dari Padang juga Bapak Nurul Huda, ladies and gentlemen. Jadi pulang kampung ya, Pak ya. I hope you enjoy your time in Padang today. And the Magister from Institut Pertanian Bogor, seperti juga Pak Dadang dan Bu Aras mungkin, dan Pak Sahab. And for doctoral program from Universiti Kebangsaan Malaysia. And the publication, some of publication from Web Index, H Index 22, citation there are many, more than 1,800. Scopus Index also very high, and Google Scholar as well. So we are very happy to have the presentation of Associate Professor Nurul Huda. The time is yours, sir.
Assalamualaikum and good afternoon to all of the participants. I would like to present my gratitude to the Entomological Society of Indonesia, to the President Prof. Danang, and also to the Chairman of the ICC ASI, Dr. Huda, for the invitation to me to share some idea related with my field on the food science and technology. So before I start my presentation, please allow me to introduce a little bit about myself. Currently, I'm working with the Faculty of Sustainable Agriculture in Universiti Malaysia Sabah in Sandakan. I think maybe some of you will be not really familiar with this kind of the university. So this kind of university is located in the north part of the Kalimantan, the top of the Borneo Island. So my campus is in Sandakan. It's dedicated only for the Faculty of Agriculture, Faculty of Sustainable Agriculture. The main campus is located in Kota Kinabalu here, the capital city of the Sabah. So Sandakan is more close to the Tawau and also to the IKN here. So it's a very big very big faculty with quite complete facility for the in the area of the agriculture. So we offer 3 programs: horticulture, crop production, and livestock production. So currently I'm working part of the livestock production stuff. Then I also want to use this opportunity to encourage any collaboration with all of the participants for the research, for the publication, or anything for the academic matter for the future. So because I think it's good if we also use this opportunity to know each other and also to strengthen the collaboration and networking among us. So actually, as mentioned, I'm graduate from Indonesia University, from Bung Hatta, IPB, and also from then for PhD I go to UKM. And then I worked here until 2005, and then I moved to Malaysia, working with University Science Malaysia for 12 years, moved to another university, University Sultan Azwainin for 2 years, moved again to University Malaysia Sabah, in Faculty of Food Science and Nutrition, as mentioned by the moderator. Actually, my field of interest is on food science and technology. And last year, I moved to University Faculty of the Sustainable Agriculture. But whether I work with the Malaysia University, I still pay attention to the university in Malaysia. I am the adjunct professor and Nutrita for 1 year. Last year, I still work with the Intera. And for this time being, I'm the visiting professor in Universitas Brawijaya. I think next week, I will go to Brawijaya for 1 month to conduct my program as a visiting professor there. Okay, here's as mentioned, my field is more on the food science and technology. And then for this time being, I put some attention for the alternative food product, include from the insect as a alternative future food. So I mentioned currently I work with the Faculty of Sustainable Agriculture, and last year I listed as one of the best 2% world scientists by the Stanford University. So I think most of us are aware about this one. And by 2015, the UN already released the 17 Sustainable Development Goal is to answer to call for the action to end the poverty and hunger, and also to protect the planet and ensure the inclusion, peace, and prosperity for all. And then one of the factors that really significantly contribute to this SDG is belong to the food and agriculture sector. Then food and agriculture sector is contribute to many of the SDGs, include for the, because they're interconnected, contribute to almost aspect of the economy, environment, and society, from hunger, malnutrition, desertification, sustainable water, loss of biodiversity, to overconsumption, obesity, and also for the public health. Meaning food and agriculture will play important part for the achievement of the SDG. We cannot separate we cannot neglect the contribution of the food and agriculture sector for the achievement of the 17 SDG as stated by the UN. I think I better here. Okay, sorry. Maybe I better here. So I think most also are familiar with the 17 SDGs, and then almost half of the SDG correlated with the food and agriculture sector. So no poverty, because mostly put in my mind, what people might in mindset of the people, that farmer, that fisherman is belong to the poor society, to the poor people. And also we need to understand juga the objective of the agriculture to provide the sufficient amount of the food product to end the zero hunger. To make sure there is no the zero hunger can be achieved, and also to provide the nutrient for the human being, good health and well-being. Also to make sure that doing the responsible production can be achieved. To also correlated with the climate action, life below water, and also life on the land. What the part that I need to focus during the presentation is the achievement of the SDG number 2, zero hunger, because one of the points that highlighted by the by the SDG number 2 is a 2.4 is a sustainable of food production. Include in this part is the alternative food for the future. Because I mentioned in the SDG number 2, the aim is to end hunger, achieve food security, and promote improve the nutrition and promote the sustainable agriculture. And the alternative so of the food material with minimum impact to the ecosystem is need to aware. We need to think about the alternative food material for the future in order to achieve, to promote the sustainable agriculture, to make sure we can achieve food security and also to improve nutrition. Because the insect, as mentioned later on, is also consists the high protein content, very good quality of the fatty acid and some mineral. So maybe because I think as entomology we are aware that insect species estimate around 6 to 10 million. Then among the 6 to 10 million, it's only around 1 million maybe that clearly that already currently well described, still more than need to explore by all of the entomological, because only 10%, maybe only 10% that well described. No, banyak lagi a lot of insect species that need to explore by the entomologists one. Then the use of the edible insect is a long history in China especially, and has been consumed for more than 2,000 years, meaning some people are already familiar with the consumption of the insect as alternative food material. And then it's estimated around more than 2,000 insect species. Maybe we're not aware, maybe because we're not familiar, it's already consumed by 2 million people in more than 113 countries. So meaning the insect already contribute as one of the source for alternative food for the people for longer time. And then some countries report the insect as part of the traditional diet, in China, Japan, Thailand, especially India, Papua New Guinea, and also South Korea. In Africa, Zambia, Malawi, Nigeria, Angola, Congo, South Africa, and also in Zimbabwe. So Oceania, Australia also reported they also consumes use the insect in this traditional diet, and it also found in the Latin America, such as Mexico, Colombia, Brazil, and also. So the term suitable for the use of the insect as food material is entomophagy, but insectivore also introduced as an alternative term for the use of the insect as a food material. And then, actually, the FAO of the United Nations endorsed edible insect as alternative for the diet for the people for the all on the world. So edible insects such as grasshopper, cricket, mealworm, caterpillar, and various lot of insects can serve alternative meat source because they contain good quality of the protein, so the fats one. Then consumption of edible insect can promote by 3 reasons. So why the United Nations promote the consumption of the insect as alternative meat? Because the impact on health, environment, and livelihood. So 3 reasons why UN try to promote the consumption of the edible insect for the people around the world. Then the global market, we can see from the global market increase yearly. In 2018, the global market is only $400 million. But in 2023, this year, the global market increased 3 times higher, to $1.2 billion expected. Meaning there's a growth, the demand for the food product prepared from the insect. Then most insects they consume is belong to the Colopetra, 30%, Lepidoptera, 30%, Hemodoptera, 30%, 40%, we have the Orthoptera, 30%, Hemiptera, 10%, and then small amount from the Isoptera, 3%, Odonata, Diptera, and other less than 5%. Is the main contributor for the insect as alternative food is belong to the Coleoptera, Lepidoptera, and Hymenoptera. I think, and then the density, the density of the insect, as mentioned, insect is available around the world. But for the some country, they have more edible insect compared than other country. So in China, in India, in Mexico, and some part of Africa, also in Thailand, they have a more edible insect species compared than other country. And then, in general, insect species contribute among half of the species in the world. And then people from the Asia and Africa is already familiar with the consumption of the insect as alternative food, more than people from America, Australia, and also Europe. Europe look like not really familiar with the culture for the consumption of insect as alternative food. So we can see this one, the growth, the market value of the edible insect is increased yearly. They increased for 3 times if you compare than 6 years ago. 6 years ago, it's only around $4,000 million, and now it's can reach around 1.2 billion US dollar. So, as mentioned, the most contribute for the insect as alternative food is belong to the beetle, Coleoptera, followed by caterpillar, beef, Hemiptera, and grasshopper here, locust and cricket. Because later on we need to highlight this kind of this insect as alternative food material, especially from the Muslim. So insect can serve a solution for the future world because there are 2 reasons. One belong because there is high nutrient content. So the high nutrient content, a lot number of the report that insect consists a good quality of the protein and also good quality of the fats and also economical advantage offer the competition. If we compare with the livestock, with the beef, with the corn, with the chicken, and also with the poultry, if we compare, so economical advantage offers and commercial medicine. So insects, they are eaten can be seen as future food because they're quite small breeding. Small breeding compares and conventional one, compares and livestock. And then high fecundity rates, low greenhouse is low pressure to the environment and ability to some space to convert the waste material to more better product, and also high feed conversion rate is a benefit for the rearing of the insect as alternative food. Then if we compare this one, so for the insect only need 1, but for the livestock is need more is need 10 for the feeding material for the low conversion feed conversion ratio compared to animal livestock. And then also for the use of the water, low use of the water, high fecundity, and also higher edible meat compared than livestock. So actually, for the insect, we can consume around 80% of the body part of the insect. Compared than beef or animal, that may be only around 40 to 50% from the body part of the animals one. And also, we compare for the cricket, part of the insect, it can consist high protein compared than livestock. So, and also little bit lower amount of the fat compared than animal livestock one. And also the use of the less use of the water. So the amount of the water needed to produce 1 pound of meat for the cricket from the insect is only 1 gallon water compared than for the livestock that need 2,000 gallons of water for the equal number of the meat one. So also low, very low greenhouse emission compared than livestock. So meaning this one low impact to the environment, more benefit for the environment. Besides, can provide the high nutrient content. And then we can see this one. So the benefit for the consumption of the insect because they provide the more nutrient, less impact to the environment. And then also for the benefit because price little bit cheaper, but it depends to the insect, to the type of the insect that the market is one. And then they use the term of IBF. IBF meaning insect-based food. It's a study that conducted in France to know what the perception of the consumer for the insect-based food. So most of them, they believe that insect-based food can provide more nutrient compared than conventional meat. And so the IBF insect-based food is more tasty compared than conventional meat, and also more affordable, sustainable, more sustainable compared than livestock, because the issue on the pressure for the environment, healthy, yeah, they call equal. So meaning whether they consume the conventional meat or the insect is can provide a good quality of the nutrient, and then lower, meaning that more people believe that IVF have more benefit. They put a question here, IVF have no benefit, more people say no. So meaning the IVF insect-based food, insect-based food can provide some benefit for the human being. It's a report from the last this year study in the among the consumer from France. So according to depend to the species, because different species can provide a different taste for the edible meat of the insect. Some say enter meat, the taste is sweet, almost nutty, and then for larva or duckling butter, it look like similar with the taste of the flavor of the whole meal bit. Dragonfly is similar with the taste of the fish. Cockroach is similar with the mushroom. Striped silver is similar with the apple. It's what you call some report about the sensory characteristic of the insects. And then depend, yeah. Egg of the water boatman is similar with the caviar. Caviar is egg from the fish. Is very important. It's very expensive for the caviar. And caterpillar is look like that is similar with the herring. Is the identification of the taste and flavor from the different species of the insects. So meaning there in Thailand, in India, in China, something like people are already familiar with the consumption of the insect. And then, as mentioned, insect can offer a different taste, different flavor, depend to its species. So, and then as mentioned previously, that insect consists of high nutrient value, high protein content, and also consists polyunsaturated fatty acid, especially for the omega-3, alpha-linoleic acid, APA, and also DHA. So we can see this one, the protein content is quite high, can be from 14 to 80%, depend to the species. So depend to the species of the insect, but in general, the insect consists high amount of the protein and also some of the essential amino acid. And then we consider with a lot of data that prove that insect meat can provide the high protein content.
Excuse me,
So,
Prof. Maybe the time is.
okay, so
Please consider,
skip.
little bit
Yeah,
more.
this one is just to mention that is a protein on the insect, so consists the essential amino acid and also the omega-3 fatty acid, yeah, alpha-linoleic acid, APA, and DHA. So what is the challenge for the utilization of the insect in her food product? Because challenge is the technology regulation and consumer attention. Maybe here there's consumer attention. Other factors, social and cultural norm, visual characteristic, awareness benefit, social demographic, food neophobia, and familiarity of the customers one. But despite the increased interest of entomology, consumer in West Europa and some part of the world still keen on eating the edible insect, because it's not familiar for them. I think I just need to skip the evidence. They try to compare the compare between the customer from the China and from the Germany. They comment the response to the insect based food. So I think maybe what I need to highlight again is some sample how the strategy. So the strategy to use the incorporating invincible insect. The strategy to the use to increase the acceptance of the insect-based food by reducing the visibility of insect in the product. Meaning they try to hiding the insect from the product. And then such as like that, they put in the insect as a part of the snack insect, and then they put in the part powder form insect pasta in the powder form, and also put the protein bar, put the insect there, and also for the burger from the insect. think I just also this one different try to introduce the insect from different food product in order to increase the acceptability of the insect food. Give me another 2 minutes. I think I need to highlight again. There are some effort in order to increase the acceptability of the insect in different food product. Say if they put the invisible and then the activity is more higher. If not compare, then if the use for the visible between the visible and invisible. So the invisible mealworm put in the product is receive higher acceptability comparison if the customer can identify that in the product there is a mealworm is there. Something like that. I think maybe I can pass, but why I need to highlight also. But even there is there are some benefit of the insect as alternative food. We also need to aware about the risk of the insect consumption. There are some issue about the microbiology, subhazard, chemical, allergic, and microbial that we need to aware before we suggest to the people to consume the insect-based food. This one, I think, regulation. There are some regulations that encourage for the consumption of the insect-based food from the European Food Safety Association. I think this one from also Switzerland. They encourage consumption of the insect. And United Kingdom is a what do you call it, the species that generally used for the insect-based product. I think this one is more important for us in Islam. What actually is good for us in Islam? According to in Islam, only locust grasshopper is clearly permissible halal to consume because in the problem consume the grasshopper. They are evident that in Islam only grasshopper clearly permissible to use for the consumption. And then Indonesian Ulama Council atau legal pond fatwa state that grasshopper and cricket is a halal. Grasshopper and cricket is halal, and then cricket can be bred for cosmetic, medicine, food. They are not harmful for the human. Meaning we can consume the cricket as and grasshopper. But and then is what do you call it, the fatwa or the Majelis Ulama Indonesia regarding the consumption of the grasshopper and cricket. And then, but a different country may be different regulation. So in Malaysia, the one is the insect that normally consumed by the people in for Sabah and Sarawak is stated is non-halal for the Muslim people as well. So the ulat sago, ulat molong is considered haram, forbidden by the according to the regulation from the Majelis Agama Islam Malaysia. Yeah, she's in halal. So meaning for the ulat sago is not permissible consume among the Muslim from Malaysia as well. So this one we need to go to come with more discussion because we have a different opinion among the Muslim scholar. The Hanafi school, you can say so prohibited, haram. But for the Maliki, maybe allow the consumption of all the insects. For as long as they are not harmful to the health and the concept is addressed to a cultural. So meaning between the Hanafi school and Maliki school as some different for the consumption of the insect as alternative food material. But for the Shafi'i and Muslim and Hanbali is a mix selected for the Shafi'i school and the Hanbali school. They both adopt a selective approach. It depends. So separate approach for the consumption of the insect. So whether it's halal or haram is one. So according to the Udin, prohibiting of the consumption of all the insects without any except is technically error and fallacy of their generation. So meaning we cannot easily to generalize one of the insects as haram. We need to come with more discussion in order maybe to see what is the benefit, why Allah provide insect for us. Maybe with some purpose and then we need to come with more discussion before we decide whether this kind of insect is belong to halal and haram. So proponent of entomology, how do Islam jurists to consider scientific evidence institution benefit, and some believe this may warrant stand to the Islamic legal position for the consumption of insect.
Okay.
And then, therefore, any insect that contribute to immunity, safe and human health and cultural assistance has met the equalities of the Thai food, Taiban, yeah, halal and Taiban, is maybe considered halal, but need to discuss again because it's not many discussion is available for the consumption of the insect for the Muslim people. I think I got conclusion. I got
Yes,
conclusion.
Prof.
Go conclusion. Need religion guideline for the concept of insect, especially for us as a Muslim people. I think it's all. Thank you because the detail of the present is available in the book Future Food. I wrote one chapter together with my former student. Also talking about the insect-based alternative. Yeah? And then a lot of number of the textbook and journal is available with promote the insect as alternative food one. Okay, I think thank you. Sorry for
Okay.
delay.
Thank you very much, Professor. Yeah, very interesting. Edible insect for food, future food. Yeah, and very complex information from Professor Nurul. Explain already about the halal or not halal status or some consideration about how to eat or to use some insect for food. Okay, very interesting. But the question will be in the next part. So the next speakers, we'd like to invite Mr. Marman Maulana from PT Prof. Pheromone. Yeah, and maybe the CV we can see here. The research interest of Mr. Maulana is rice crop pest control in rice, and the expert and skill as well about rice crop will control and insect control as well. And Mr. Maulana is graduated from IPB University 2010. And then it is the time for Mr. Maulana.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Good afternoon, ladies and
wabarakatuh.
gentlemen.
Good afternoon.
Can you show my presentation, please? Yeah, I'm from Prof. and our company basically work with the pheromone. So, have you heard about pheromone before?
Yeah.
Yes, right? So if you open the social media or the e-commerce, there's a lot of advertise about the pheromone perfume, right? So they promote it to attract male or female. While our company work with opposite. We use pheromone for mating disruption. Not a mate, but mating disruption. Yeah, it's opposite with the other pheromone we found it in market. So today I will talk about the development and adoption of mating disruption technology for sustainable management rice to control yellow rice stem borer in Indonesia. So it is I want to show about how female release pheromone sex in the field. Where's the?
Is it possible to play?
No.
Is it movie?
No, it's just picture.
Okay.
Yeah, here is the female moth when release pheromone sex in the field. It's natural in the field. While when we put our product here, they will release pheromone cloud in the air. So the male moth will be difficult to find female moth in the field due to the high concentration. So it can be cause the mating disruption and prevent male to mate female. And it can be reduce pest population season to season. It's not drastically down, but it can be like season to season. So why mating disruption? First one, if you remember what Prof. Dadang mentioned in the morning, insect or pests cause the high damage in the field. Farmer lost a lot of ton of yield. Also, they lost a lot of money to control pests in the field. Second one, to meet with world population growth, we need to prepare a lot productivity. In some area, they have a target to get 8 to 10 tons per hectare. So we need a healthier crop to get high yield and meet with the target. The third one, they have an insecticide resistant or tolerant issue for some molecule to control some pests. And already happened since, if not mistaken, since 1960 something, we found some resistant issue. And time to time, some molecule was tolerance or resistance. And the next one is very costly and takes time to prepare new molecule to control pests. And method exception will be like part of IPM strategy because will be reduce chemical application and save for beneficial insect in the field. So how mating deception work? Like I mentioned before, here is the natural male and female mate in the field. Then when we put our product, we call it dispenser. Maybe it's like, what, dispenser? Like a gallon water or something, but our product, we call it dispenser. When we put here, the male will be difficult to find female. How? Because it will be like false trial and following, then camouflage because the high concentration of the pheromone cloud in the air, desensitization of males to mate with female, then the sensory imbalance. In fact, there will be create mating disruption, prevent male to mate the females, then it can be suppress pest population in the field. Yeah, in Indonesia, Prof start study or trials since 2018. We test with the formulation early stage, then going to the 2019, we start the final formulation there. We got the what's the dose rate. Then starting 2020, we believe and we already got confidence to register our product in Indonesia. 2020, we create a lot trials and demo in Indonesia. And also we got approval from the minister in 2021. Then 2022, we also continued our activities to create the demand and we launched the product in 2022. So what's our commitment to develop mating disruption technology to control WSB in Indonesia? We have done to make a study in more than 115 locations in 56 regency in Indonesia. It's not a lot, not small number. We conducted the trials in more than 1,500 hectares in 4 years. We contribute or partnership with more than 2,000 farmers. And we are the first company to launch the product in Asia, not in Indonesia, but in Asia for the mating disruption to control yellow stem borer. Make applause, please. Yeah, we can release the condition not too tight, yeah. So what is our parameter to proven our product? The first one, reduce the infestation and damage in the field. Second one, how to reduce the insecticide application frequency. then observe the healthier rice crop, because at the end, the farmers will be calculated with the yield or the money that they got after harvest. So based on that, our study on 4 years, we believe our product can decrease or reduce 40% damage in the field. What is the parameter? We also make a assessment with the calculate the damage in the field compared to the healthier crop. Then also we monitor the pest population in the field. We use the sex pheromone to know about the number of pest population in that location. If we see the zero here, means that we compare to see that these two different pictures. In the left side, we see that so many moths captured here. Means that high pest population on that area. And when we see on the other side, it's opposite. Looks as zero. Means that no pests on that area. Means that the product like works because no more we found it in the field. Next one. Based on our study, more than 55% reduce the number of insecticide application. And we believe when we reduce the number of insecticide application or chemical insecticide application, so we can give the more benefit to the environment and also for the applicator. Not only talk about the damage reduction, but also we talk about the healthier environment and the healthier applicator and the farmers. Yeah, and for the how to know about the contribute to healthier crop. Based on the trials, more than 74% yield within rate. Means that from the all location that we conducted the trials, almost like we got the higher yield than the CGP or commercial grower practice. Means that the grower, when they use chemical insecticide to control YSD to their area. So based on that, in average, netting disruption provide high yield around 0.4 to 0.5 ton per hectare in average. It can be like high or low than 0.4, but in average around 0.5 ton per hectare. So what we are doing to give the activities to the field to create the demand. Like I mentioned before, our trial is not small. Our plot minimum 4 hectares. We install the product in 4 hectares per plot. If we calculate with the number of the trials, a study will be wide area. Also, we have networking with the stakeholder and growers in the field. They must have the value to the farmers, to the growers, to the government in the location. We discuss, get the impact, feedback from them, how to improve product, how we deliver our technology. Then we spread the word with brand and value, with partnership or collaboration with the others company in Indonesia. I know this is not the easy one because it's new technology. The farmer will be not easy to adopt our product, how to implement in the field. So we still make some activities to leverage our information technology to the grower in Indonesia. And we believe with our product, we can help farmers, we can help environment, and we can help all of us, especially in Indonesia. So what's the benefit of methane reception? The first one, like easy to install because we just only one bamboo sticks. No need the tank, spray tank or other thing, just need bamboo sticks and zip ties. And it can like reduce plant damage, reduce number of the spray WSB insecticide. It's contributing to the healthier crop, safe for beneficial insects, and no residual. We have a test and collaborate with PI in 2021 to get the residual in the grain yield. There's no plant toxic effect. And this product is specific target species. Our product just to control WSB or yellow stem borer and striped stem borer or Hylos persalis. I think, yeah.
Okay.
Yeah, just to slide. Go.
Okay.
You promised 10 minutes, right? Yeah. This is our product who is registered in Indonesia under brand Alfium, and we collaborate with Syngenta Indonesia to leverage, to promote this product in Indonesia. So, and thank you from my side, collaboration with Syngenta Indonesia, and I'm also with collaboration with BB Buddy, IPB for when we develop this product, and all farmers in Indonesia who collaborate with us. Thank you, and Assalamualaikum warahmatullahi wabarakatuh.
Thank you. Okay, thank you so much. Very interesting, ladies and gentlemen. All of the presentation is very interesting. The last one is about pheromone that could be arranged then for insect pest management and very safe, according to Mr. Marman. Okay, actually we already find our coffee break time. So please be free to have your coffee break before I give to Master of Ceremony. But before that, let me use a little bit, maybe around 5 or about 5 or more minutes to discuss with the speakers. If ladies and gentlemen have something that you want to give to the speakers, a feedback kind of question or opinion, 1 or 2 people, and with a very short question, if possible, to raise your hand and give the question from now on. There's a time. Okay, sir. Maybe please give some mic, a mic, and then please introduce your name and deliver the short question, please.
Okay. Thanks for the time. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
My question for Professor Dr. Nurul Huda.
Yes.
Okay. I'm sorry because I didn't attend from the beginning. So maybe my question already explained in your presentation. Related with the use of insect for
food. Yeah.
You know that 1 or 2 years ago here in Indonesia, we had a problem with the shortage of oil, with the vegetable oil. So what is, in your opinion, the possibility and the feasibility of using oil? Using the use of oil extracted from the insects for that use. I mean, for
oil,
let's say for cooking or maybe any other functionality from this kind of oil. I think that's my question. Thank you.
Okay, thank you so much. Please, Mr. Nurul Huda, maybe what kind of oil? Maybe what kind of insects oil? Maybe you can specify. Any kind. Okay, sir. Maybe please short answer, please, because on 15:50, 15, 15:15, we will have a hybrid Zoom. Okay.
Okay, thank you. I think you're talking about the fat or oil from the insect. As mentioned, according to some report, the the fatty acid from the insect is consists also the polyunsaturated fatty acid, especially for the omega-3 fatty acid. This one is quite interested because normally for the omega-3 normally is only available from in the marine product. But according to some journal report that omega-3 is also available in the insect meat. This is I just want to this thing that need to highlight for the benefits help from the consumption of the insects because we can get some omega-3 from the insect meat. I think it's all. I think for this time being, for the answer of the questions one. But please, we can discuss more detail because I know the number of the research are related about the insect oil or insect fats. Okay, thank you.
Okay, thank you. I hope it's enough answer. Okay, please, sir.
Yeah, thank you. My name is Andy from UGM. I have a question for the last presenter. Actually, when is the product installed in the rice plant for having a coverage for the whole season? Thank you.
Is it clear? Okay, please.
Thanks, Prof. Andy. Yeah, the great question. Yeah, basically to install our product in the field, at least we need based on our study, we need a 4-hectare plot with 20 dispenser per hectare, and we just install one time per season. So based on our study, we have also another, in parallel, we have a release rate study. And when we install the product can be stable in at least 4 to 5 months in the field. So they still release the pheromones until 4 to 5 months. I mean, that will be covered for one season rice field.
Okay, sir. Thank you. Is it enough? Okay. And the last question, if any, yes, sir. Any mic to Mr. here in the middle of the area? Okay.
Thank you. Good afternoon. I would like to ask question to Dr. Wahida. That's such a really nice and well-detailed information. For me, this is a new method by using a part of organism to control a pest. However, I would like to ask you, how do you infect the pests, the methods in the field itself? And as you presented in your presentation, they are able to infect multiple kind of insects. So what would happen in the field once we apply the Wolfar to the targeted pest, and once that pests are predated or parasitized by other kind of beneficial insect, whether that Wolfar could affect these beneficial insects that are present in that field? Thank you.
Okay, ma'am. Please, this your answer, please. Okay.
Thank you for the questions. For this preliminary study, we injected the Wolfar into the adult RPW. That's why you can see the results from what I presented. Regarding the pollinators in or beneficial insect, either they will be affected with Wolfar or not. I think further study needs to be done, and with this Wolfar synthetic peptide, we still need to study the genes related with the surface protein so that it could be really specific and attack on the RPW. Thank you.
Okay, thank you so much. I guess, ladies and gentlemen, we already have 3 presentation from our 3 speakers with very valuable topic, and also we already have discussion with 3 also question from the audience. Thank you so much for all the speakers, Dr. Wahida, Dr. Nurul, Professor Nurul, and Bapak Marman Maulana. Thank you so much for your presentation. We hope we can continue to share the knowledge to others. Thank you so much. Please give applause for our 3 speakers today. Thank you so much, and I will close with Alhamdulillahirabbil alamin. Assalamualaikum warahmatullahi wabarakatuh. Let's go to the next session.
Once again, let's give an applause for the plenary one of this international conference. But before that, we hope that for the invited speakers and moderator to take the position in front of the stage, and we will see the token of appreciation from the committee to the invited speakers and the moderator. We invite the moderator and invited speakers to take the position in front of the stage. We would like to invite the presidents of Entomological Society of Indonesia to take the place and give the certificate, and also the chairman of this international conference, please. First, the certificate for Miss Dr. Nurul to take the position, and take the certificate from the presidents of Entomological Society of Indonesia. Let's give him applause, ladies and gentlemen. The next one is the token of appreciation. This is the amazing moment, and thank you so much for Dr. Nurul to take the joining us here at the international conference. Next, for Professor Nurul, please. This is the certificate from the presidents of AC, and also the token of appreciation to take the position. All right, this is the moment from the chairmen of the international conference. Let's give an applause, ladies and gentlemen. And the last one, the certificate for Prof Incorporation delegation. Thank you so much for your time in this international conference, and this is the token of appreciation from the chairman of this international conference. After this, we take the photo station, and also the certificate appreciation for our moderator now is Dr. Heni Herwina, the chairman of Entomological Society of West Sumatra, please to take the certificate from the presidents of AC. Ladies and gentlemen, next is photo session, please, to Professor Dadang to take the position, and also the invited speakers and moderator. Once again, let's give an applause, ladies and gentlemen, the amazing conference in the plenary one of this international conference. Ladies and gentlemen, before we start the second plenary of this conference, before that, please to take the food and drink and also enjoy your coffee break time, dear ladies and gentlemen, in the 15 minutes to you to take the food, drink, and also in the coffee break time. Enjoy. Also, ladies and gentlemen, we would like to inform that the semi-final of Linear Games 2 will be start. And for the participant, please be prepared and go to room A with the Dr. Rina Rahmawati as your licensed officer leader. Good luck and see you for the semi-final Linear Games 2. Ladies and gentlemen, in a few minutes, it's 15 minutes, we will start the plenary 2 of this international conference with the invited speaker that has joined us virtually. And before that, ladies and gentlemen, to enjoy your drink and food in this coffee break time. Ladies and gentlemen, we invite you back to the ballroom. Please entering the ballroom, and in few minutes we will start the plenary tour of this international conference. Bapak Ibu yang kami hormati, kami kembali mengundang Bapak Ibu untuk dapat masuk dan menempati tempat duduk di dalam ballroom karena sesaat lagi sekitar 10 menit kita akan memulai kembali International Conference plenary kedua. Terima kasih. Bapak Ibu yang kami hormati, kembali kami mengundang Bapak Ibu untuk dapat menempati tempat duduk di dalam ballroom karena sesaat lagi International Conference plenary kedua akan segera kita mulai. Ladies and gentlemen, we back to invite you to entering the ballroom because in a few minutes, it's 5 minutes, we will start the plenary two of this international conference. Ladies and gentlemen, we start the plenary two of this international conference. But before we start the conference in the plenary two and also with the moderator, we have to say thank you so much for all the sponsorship and partnership. Badan Pengelola Dana Perkebunan Kelapa Sawit BPD PKS, Prof Incorporation, PT Bayer Indonesia, PT Syngenta Indonesia, Corteva AgriScience, Crop Life Indonesia, PT Sanjoy Agro Indonesia, PT Tangguh Agro Unggul, Alister Indonesia, Crop Care Indonesia, PT Astra Agro Lestari, PT Agricon Indonesia, Asosiasi Produsen Pestisida Indonesia, Peladang AgroScience, PT BC International Tbk, PT Petrosida Gresik, and PT Inti Every Spring Indonesia. Thank you so much for your support, and also we have to say welcome and thank you so much for all the participants that has joined us virtually, and we will goes to the plenary two of this international conference. Ladies and gentlemen, we move to the next agenda, the plenary two, with our invited speaker that has joined us virtually to leading the session. Please welcome our moderator, Dr. Purnama Hidayat from IPB University, Indonesia. Please giving applause and time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, in the next about 75 minutes, we will have a special session that is the online keynote speaker presentation. In this special online session, there will be 5 speakers. They are Professor Damayanti Buchori of IPB University Indonesia, who is currently she is in United States of America. The second speaker will be Dr. Susan Kalumpang of UPLB Philippines. And the third speaker will be Dr. Katsuyuki Eguchi of Tokyo Metropolitan University, Japan. And the fourth speaker will be Dr. Kris Wyckhuis of University of Quensland, Australia. And the last speaker will be Dr. Srinivas of PT Bayer Indonesia. Every speaker will have approximately 10 to 15 minutes presentation, and the discussion will be done after all speaker presentation. Before presentation, I will read a short CV of each speaker. And for the first one, I will read short CV of Professor Damayanti Buchori. So do we have the CV? Okay, this is the short CV of Professor Damayanti Buchori. He is from IPB University. Her expert is in entomology, evolutionary ecologist, transdisciplinary and sustainable science. P.hD from Department of Plant, Pests and Diseases, IPB University. Her magister in entomology, University Illinois at Urbana-Champaign, USA, and her doctoral in evolutionary ecology, Indiana University at Bloomington, USA. Dr. Damayanti, are you ready?
Yes, can you hear me?
Okay, so
Okay, yes.
Budami, you have 10 to 15 minutes and
Okay,
time is yours. Anyway, where are you now, Budami?
I'm in Minneapolis right now. It's cold
Okay.
and it's 3 o'clock in the morning. So,
Okay, Budami,
okay.
time is yours.
Thank you so much. Assalamualaikum warahmatullahi wabarakatuh. Selamat sore. Good afternoon for everyone. And Pak Pur, if you can remind me like 3 minutes before the 15 minutes, then I will make it faster because I'm afraid that I made too much slide, but hopefully I won't be too long. So allow me to share the screen. Okay, I hope you can see. Oh my God. Wait a minute. Let me minimize. Okay, so the title of my talk is Future Entomological Research in Tropical Archipelago Ecosystem, Integrating Conservation and Agricultural Entomology. So this was actually asked by the organizer, whether I can discuss about the future research in Indonesia. So coming from a background of evolutionary biology, I'm trying to integrate what is it that is important from the ecological point of view toward agricultural entomology. So maybe this is a bit biased to my field, but there are so many research in entomology. So of course I cannot cover all of it. Okay. So, okay. If we look at the research overall, one can see that the history of entomological research in Indonesia actually started from the colonialism era during the Dutch occupancy. And we can see that there are a vast array of research that has been done in Indonesia, whether it's the taxonomy, biology, pest control, etc. So there are what you call the classic research that have been done in the country for the past, I don't know if we count since the Dutch occupancy, maybe it's the last 100 years or so. And we can also see that in the future, there are advances in research that seems to be more interdisciplinary. For instance, the landscape ecology has now been integrated a lot into entomological research. I know Professor Andi is also working on that. And then also this new approach of using endosymbiont, Wolbachia, to control mosquito, and also the Chinese researchers are using Wolbachia to control the brown plant hopper. So that's another venue of research that seems to be the latest development. And of course, the nanotechnology, remote sensing, AI for pest management. So there are many, many advances in research that different institutions are now conducting. If we look at Indonesia, there are actually many opportunities for research topics to be conducted in the country, because of course the country is large and we have many different areas that are interesting to be researched upon, very diverse, biodiversity country. And also the opportunity for collaboration, either both local, national, and international, is also quite open. Of course, there are many challenges. Maybe the numbers of researchers are not that many, and the interest to the research, it needs to be increased. And of course, funding is always a problem. And research policies and research atmosphere is something that need to be increased in the country. So with that background, I would like to focus on the next 10 minutes or so, where I'm dividing the presentation into 4 parts. The first is looking into the past. What have we been doing in terms of research in Indonesia? But I'm just focusing on the last 20 years. And then I'm putting into context the tropical ecosystem. And then the third one is cutting edge research in ecology. And the last one is more about the future entomology research. Okay, so what have we done in the last 20 years? So this, I tried to research what has been done in Indonesia. This is, I was helped by my student here to collect data about 1,864 articles from JAE, JHPT Tropica, GPTI, and also Scopus Index journal. So overall, this is what we can see, that the numbers of research in entomology have increased over the past 20 years, especially since the last 5 years, it doubled. So we started in 120 and now we have increased to 900. Almost 60% are focusing on agricultural entomology, agricultural pests, with some focusing on also on urban pests, and the others, you can see that up in the slide. And then the topics of the research is pest control. 46% are focusing on the pest control. And if taxonomy, which should be a lot, because Indonesia is a mega diverse country, there's only 15% who's focusing on the taxonomy and identification. The biology of different insects is also quite, number is the same with the taxonomy, the percentage. And if we look at the pest control, what are the topics of the pest control? And it seems many are related to chemical control. And the next one is biocontrol. And there are the others, physical, resistant, using for resistant variety, IPM, etc. We have seen a new influx of molecular techniques that is being used for insect-related research. People start using mitochondrial gene rapid, also using genetic variation, using ITS1 and other genes. So it's getting more attention right now using the molecular techniques. And of course, the smart agriculture. Of the 1,800 something topic, there are 20 articles that is focusing on the smart agriculture. Either it's about internet-based control, satellite spectral data analysis, expert system, etc. So we are on the right track. Okay, so now I'm going to contextualizing what I think is important about conducting research in Indonesia, particularly if we understand that Indonesia is an island country. So I think one of the major issue in tropical archipelago ecosystem right now is still land use transformation, habitat degradation, species loss. This is still a major issue in the country because we know deforestation is still going on, and we still don't know how actually many species are there in Indonesia. And thus, we never know how many are lost because of the deforestation. And the second issue is about understanding what is meant by island. So here I would like to introduce the island biogeography theory, where it says that the species diversity within an island is depending on the size of the island and also the isolation of the island from the mainland. So endemicity is usually high in island, and that's why in Indonesia we have diversity because of the high endemicity is also very important. And one that has not been looked at is actually what is the implication of this island biogeography theory for pest management. And the last one is about emerging pests or the invasive species. And so this is putting into context the next, the third part of the presentation, which is a cutting edge research in ecological entomology. So I'm just going to focus on two big issues. The first one is land use transformation and its effect on diversity and also looking at pollination. And the second one is the archipelago ecosystem, how to use island biogeography theory to understand ecological process in small island, particularly as it relates to biological control. Okay, so land use. On this left side, you can see that deforestation in Indonesia is quite fast. In this past, okay, since 1985, we have a data here from WWF that we have seen how Sumatra has been transformed into now we only have 24% left of the forest and maybe even less. So one of the important issue to study is about land use transformation. What happened when land use has been transformed from rainforest to jungle rubber, and also all palm. So the next is going to be several slides of the research that we conducted together, Pak Purnama and I. We belong to an effort project. This is a collaboration between IPB, Jambi, Tadulako, and also University of Göttingen. So we have collected different insects in different years. Altogether, we have 4 collection years, 2013, 2017, 2021, and 2022. So overall, we have collected more than 1 million. And the data down here is specific, just the data for the 2013. So when we look at the number, at the composition of the taxonomy diversity that we have collected, the data shows that the most abundant are the Isoptera and then Hymenoptera, and Collembola and the rest. Okay, so what does the data say? So when one look at the transformation of land use, okay, F is forest, J is jungle rubber, R is rubber, and O is all palm. One can see that for ants, there is a decrease in abundance per meter square from the land use transformation, with forest and jungle have the most abundant, is also most species rich. And usually the rubber and the all palm is usually the last. So it's very clear here what is happening in ants. And when you see the composition here on the right side, it seems that the forest and also the jungle, the composition is quite similar, but the all palm is totally different. And also the rubber is totally different. So this is from the ants' point of view. If we look at the spiders, then we see the same trend. It's always the forest is more, and the jungle rubber is most abundant, species rich. And it's always the rubber and the oil palm that is very poor. And also the composition is the same. So the ants and the spiders have the same composition. The beetles, more or less also the same, although the intensity is quite different. And here we also collected biomass study, and this is using isotope, which I will explain later on. But here the species richness is also different between the land uses, and also looking at the composition. This is the composition between two different landscape, and one again can see that the jungle rubber and the forest is similar, but the oil palm is always very different.
Hudami, you have 3 more minutes.
Oh my God. Okay. Okay, now I'm confused, but okay, let me finish. So let me just skip on this one, because basically this is just saying that looking at the habitat fragmentation, one can also see that it affects the pollination. So with the increase of the land intensity, you have a decrease of pollination abundance and pollination services. Okay. And another thing that is interesting to look is actually looking at the pollination ecology. And one can see that when you look at the pollination network, we can learn about what is the foraging behavior of the pollinators, where do they go? And so this is a research that we did looking at the pollination network. The blue one is wildflowers, the yellow one is the flower strips that we planted, and the green one is actually the corn. And here we can see that different types of bees are basically, they are going, this is a research on stingless bee, and the majority of them are going to the wild plants and also the refugia or the flower strips that are planted. So this is very interesting in that we have crops, but actually the stingless bee, they prefer to go to the wild plants. Okay, so the second part of the research is going to look at this biological control on island. Now, what's interesting about biological control is that the classical biological control theory states that if there is an invasive species, you have to put in exotic natural enemy coming from the origin of the invasive species. But what is happening is that there is what is called an indirect non-target effects of biocontrol arising from a food web subsidy. So what is that? This is a study conducted several years ago on a biocontrol agent on knapweed grass. And this research found that the biocontrol agent that should be controlling the grass is actually giving food subsidy to a rat. So you see here, there's a rat here. So there are changes in the system when you introduce a new species into the system. So this is something that we have not seen before, or we have not really looked at before. And so what I did in Indonesia, me and my team, we did that, and looking at the bioagent of C. odorata, Chromolena odorata, and its biocontrol agent of Cecidocaris. And what we found is that this Cecidocaris is being attacked by all these braconids. So there are natural enemy, local natural enemies are attacking the biocontrol agent that were being introduced. And we did this in Lampung and Bogor. We did this also in Sulawesi. And there are different natural enemies that are attacking the biocontrol agent that was introduced from outside of the country. So this posed a question that biocontrol agent is not as easy as we once thought. There are new associations between exotic biocontrol agent with local community. What is in this implication? Okay, so this is something that we haven't thought about before that is very important, especially since Indonesia has many islands and classical biological control on island is not being looked at. This is a conservation issue that is important to study. This is another research that we did on Bactrocera, and it's also being attacked by local natural enemy. So this old association, which is the classical biological control versus the new association, has actually been Hokkanen and Pimentel in 1984 already mentioned that classical biological theory should be revisited because local adaptation can actually happen in quite fast. Okay, so this is one of the things that I want to excuse me, stress, that it's very important to understand small island, the island biogeographic principles, and the food subsidy for pest management in Indonesia. Okay, the last one, I just want to use this one slide. I'm not going to elaborate on this, but vision of the future of entomological research in Indonesia. You need to understand, we need to understand tropical archipelago. Excuse me, I need to drink. Okay, tropical archipelago, small island phenomena. It's important to integrate the basic principles of ecology, conservation, and agricultural entomology. It's important to look at the community processes. This is not much studied. And so it's important for us to develop skills. And the skills that are for the future are now, which is called there's a stable isotope, there is the environmental DNA, there's a biomass study or biomass technology that is quite new. And also big data, machine learning is very important for research in the future, including coding. So people or students in entomology should learn about coding using R and Python. So I'm just going to, okay, in summary, the future needs new insights into existing problem. We need to learn about new methods and techniques, and we need to build a robust research question. That is the key for the future research. Thank you, Pak Pur, and sorry for going a bit further.
Thank you, Bu Demi. That was very interesting. And please keep it warm because you are like 3:44 in the morning. It's very early.
Correct. Thank you.
I would like to invite the next speaker, Dr. Susan Kalumpang from GPLB Philippines. I will read her curriculum vitae. His education is National Crop Protection. Oh no, her institution, I mean, is National Crop Protection Center, College of Agriculture and Food Science, University of Philippines, Los Banos, retired. And research interest is pesticide chemistry and chemical ecology. Her expert and skill pesticide residue analysis and pesticide risk assessment. Her education is B.Sc in chemistry in Silliman University of Philippines, Magister from Agricultural Chemistry University of the Philippines, Los Banos, and her doctoral from D.Sc Agriculture Chemistry, Tokyo University of Agriculture. Dr. Susan, the time is yours.
Okay, thank you. Good afternoon to everyone. Sorry. I'll be talking about plant semiochemicals for insect pest management and my research, which has focused on learning from the Filipino farmer. What do we know about this topic? We know that due to climate change, there is an increase in insect pest populations. And the pest pressure is very high, especially in vegetable production, leading to tremendous losses. Therefore, farmers sometimes do mix pesticides into cocktails out of desperation and ignorance of proper pesticide management. There may be some also who harvest the vegetables without following the recommended pre-harvest interval. So why does the insect attack the crop? Insects are attracted or repelled by plant chemicals, and these volatile chemicals are detected at a distance. These chemicals can be found on the plant surface or inside the plant. Now, semiochemicals are insect behavior modifying chemicals. These are chemical cues which affect insect behavior in terms of finding food, oviposition site, choice of sexual partners, avoidance of danger, and habitat for the larvae. Now, learning from the farmer knowledge on crop protection, we have found out that on the field, marginalized farmers do observe what is happening in the field, the pest problems. They try to analyze what causes the pest problems, and they do experiment on ways how to manage the pest. Subsequently, these can come out as innovations of marginalized farmers, farmers who have very low income, have low access, poor access to inputs, and poor access to knowledge on crop protection. And the role of the researcher is to elucidate the mechanism in the field for the innovations made by these farmers. And this is where I have focused my research on chemical ecology. So I will talk to you about the experience we had in a corn, Ipomia triloba, Asian corn borer interaction. Now, Ipomia triloba was found by farmers to repel the corn borer from the sweet corn fields. This came from an observation of low corn borer damage. Therefore, the farmers said they allowed this weed to grow in the cornfields at a certain density. It is not allowed to freely grow because it is a weed and it competes for the fertilizer. Now, Ipomia triloba is also utilized as a vegetable in the mountainous areas. This is what corn farmers believe and practice, that sometimes there are volatiles that are released by the plants, and these repel the Asian corn borer. Now, my research found out that corn plants release volatile chemicals which corn borers use as chemical cues to determine the presence of cornfields from a distance. Now, when corn is entwined with Ipomia triloba, it releases a repellent chemical, 1-methyl-ethyl propyl disulfide. And I found out that this chemical is also released when the corn plant itself is wired. So it is the stress of the effect of the weed entwining on the corn plant that stimulates the release of this repellent chemical. Now, field trials were conducted by my co-researchers, and they tried planting corn with Ipomia triloba strips within and in borders alongside corn. And they found out that corn entwined with Ipomia had less tunnels and egg masses than corn alone. Therefore, we found that Ipomia can help farmers reduce cost of insecticide use for corn borer management. Now, look at another interaction of corn, Asian corn borer, when a seaweed is used or placed on the corn plant. This is a practice of marginalized farmers in areas, in certain areas only, in certain areas in the Philippines. And in rice, it is also used by farmers in Bicol region. Now, this seaweed is sargassum seaweed, and this is usually found on the seashore after a storm. This brown seaweed is it grows on the seabed, and it gets uprooted during a storm, so it can be found on the seashores. Now, farmers collect this seaweed and place it on the corn plant along the border, and they believe that the smell of the seaweed will repel the corn borer from entering the cornfield. Therefore, strategically, the seaweed is applied from the wind direction, based on the wind direction, where the wind comes in, that is the site where you have to place the seaweed. Now, as a scientist, we did white tube bioassays, and we found that fresh seaweed had higher repellency than the dried. And this is due to the loss of the chemicals, which are volatile in nature. And so we found that fresh seaweed showed a repellency of 77%. And we tested 2-ethyl-1-hexanol, and it had quite similar effect in terms of repellency. This slide shows to you the different chemicals that were detected from the volatiles emitted by the seaweed, which we collected and which we identified by GCMS. We tested it in a white tube olfactometer, and we, as a standard, we used phenylacetaldehyde, which is a known attractant for corn borer. And when you use a control, then you're able to see that your system is working when there is a attractant effect based on a known chemical that is an attractant. And here again, 2-ethyl-1-hexanol and 2-ethyl-hexyl glycidyl ether are the most repellent of the seaweed volatile organic chemicals. Therefore, there is a chemical basis for the observed phenomenon in cornfields, which Filipino farmers observe and practice. Now, we did a few field age experiments, and we found that there were fewer egg masses laid in corn with brown seaweed at 45 days after planting, or a reduction of 38%. Fewer eggs were also observed. This indicates that the female corn borer was less attracted for oviposition on corn with brown seaweed. Now, we go to a rice ecosystem. There are areas in the Philippines wherein there is where there's this plant, a wild ginger, that grows very well in their communities. And the farmers in Infanta, Quezon, observed and used this plant, which is called tagbak in the Filipino language, or Coloracia elegans, and they use it in the rice field to repel certain insect pests. Now, we did validation trials at University, and we used the method used by the farmers. And as you can see in the picture, we placed the fresh tagbak stalks every 2 weeks in the rice field, and we observed that tagbak repelled insect pests of rice starting at 14 days after transplanting until maturity, and the GLH was lowest in the tagback treated paddies. And more importantly, we monitored natural enemy populations, which increased in the tagback treated paddies. In terms of yield, there was a difference from control, wherein we were able to get 5.8 tons with tagback and 7.3 tons for chemical treated. So therefore, for marginalized farmers who do not have, are economically at a disadvantage, who can hardly afford to buy fertilizer or pest control chemicals, the alternative of using a tagback is something that makes their system sustainable.
Dr. Susan, 3 more.
Yes. Okay. So we did bioassays, and in petri dishes, we were able to see that the male GLH did not move when tagback was present. This means less GLH mating in the field. And in the olfactometer studies, we tested these different volatile organic chemicals, and we found that alpha-terpinine and alpha-pinene were the ones that showed very high repellency against green leaf hoppers. So the use of plant semiochemicals for insect pest management in corn and rice production can be promoted in areas where plant resources are available. These plants are available to reduce dependence on synthetic insecticides. Now, very quickly, we did a study on eggplant lemongrass intercropping, and insect behavior tests showed that the eggplant fruit shoot borer was not attracted to eggplant when lemongrass was present. So we also counted the damage and for the different major pests, which was reduced when lemongrass was intercropped with eggplant. And we also found in terms of yield that there was a significant difference in plots with lemongrass compared to without lemongrass. So we can learn from our farmers, and as scientists provide a scientific basis for their innovations. Thank you for your time and attention.
Thank you. Thank you, Dr. Susan. That was interesting. Please stay online for discussion later.
Okay, okay.
Okay, for the next presentation, I will invite Dr. Katsuyuki Eguchi. He is Associate Professor in Tokyo Metropolitan University, Graduate School of Science. Her research interest is a taxonomy and biogeography of terrestrial invertebrate. His education, P.Sc in University of the Wyckhuis, Magister in Kagoshima University, and his doctoral degree in Kagoshima University. His publication can be seen in a ResearchGate and also in our ORCID 000-002-1054-1295. Dr. Katsuyuki, the time is yours.
Can you hear me?
Yes.
Yes.
Okay. So thank you very much for the opportunity to present part of our ongoing research activities. Oh, sorry. Okay. So my laboratory is focusing widely on the taxonomy, phylogeography, and ecology of various terrestrial invertebrates, as well as research on the control of invasive species such as ants. And international students from Taiwan, Vietnam, Laos, Indonesia, Nepal, Sri Lanka, and other countries and regions have so greatly contributed to our laboratory's activities. So the species names are the most basic and important known in biodiversity research and conservation. This is because biological knowledge about each organism is accumulated, managed, and distributed by linking them to species name. And the necessary knowledge is effectively retrieved using species names as a keyword for searching. So this presentation will highlight the importance of taxonomy in biodiversity conservation with 2 case studies on soil animals in a broad sense. The soil animals are thought to play important roles in terrestrial ecosystems as mechanical decomposer, ecosystem engineers, and keystone predators. So the Wyckhuis, the island chain between the southern Kyushu and Taiwan have been considered a hotspot for endemic species and lineages in East and Southeast Asia. Vertebrate and some large insects and vascular plants that easily attract social attention have been relatively well studied in this area. However, soil-dwelling invertebrates have not yet been well studied. So here I briefly explain the formation of the Wyckhuis and Taiwan. So the Wyckhuis or Ryukyu archipelago had constituted the marginal area of the eastern continental shelf, but was uplifted and moved to its present position with the opening of Okinawa Trough. On the other hand, Taiwan emerged about 2 million years ago in the process of collision between the Philippine Sea Plate and the Eurasian Plate, and uplift proceed from the north to the south. And during the glacial period or cold period, come repeatedly during the Pleistocene Ice Age. Several islands formed a large landmass due to the drop of sea level. And the islands of southern Ryukyu and Taiwan formed a huge peninsula extending from the southern China, while the Yakushima and Tanegashima Island in the northern Ryukyus were connected to the Otsumi Peninsula in southern Kyushu mainland. On the other hand, the Tokara and Kerama Strait, which divide the Ryukyu archipelago into 3 island arcs, exist permanently even during the glacial periods and have acted as a biogeographical barrier for terrestrial organisms. So the Ryukyus and Taiwan and adjacent areas with such a complicated geographical history is covered in our ongoing project. The first example study is on the large centipede genus Scolopendra. The genus is the taxon established by Linne in 1758 at the beginning of the modern taxonomy. And approximately 100 species have so far been described under this genus. So the Tsukamoto-san, my student, recently described and named a new species of Scolopendra from the Ryukyus. Due to its large body size, you may imagine that Scolopendra centipede has many morphological characters useful for recognizing species. However, in reality, there are few morphological differences among species. Furthermore, some morphological characters exhibit large variation within the species. Therefore, species-level classification is still chaotic. So before this study, 5 species of Scolopendra had been recorded from Japan and Taiwan in total. However, local people in the Yambaru area of the northern Okinawa island have said that there are large Scolopendra centipedes. And from around 2010, the presence of the undetermined centipede species has become widely known among pet traders. Since then, it was being intensively collected by them, while the natural environment in Yambaru area is being destroyed by various human activities. So therefore, in order to promote the conservation of the species and its habitat, we started a project aiming to confirm the taxonomic status of the undetermined centipede species. Because the determination of the species name is actually important when taking various formal conservation measures, as mentioned later. So because the population size seems to be reduced at the beginning of our study, we could get only few specimens for this study. So we had a very hard time in morphological examination because of poor morphological characters in this group of animals. Nevertheless, finally, it could be distinguished from its relatives from Japan and Taiwan by a combination of several morphological characters, including the tibia of 20th leg lacking a spine-like seta, and the male genitalia lacks gonopod. But these evidence may not be enough. So in order to obtain a further strong confirmation, molecular phylogenetic analysis was performed based on a total of 2,041 base pairs length dataset of mitochondrial CO1 and 16S gene and nuclear 18S and 28SS genes. As a result, the undetermined Scolopendra species was recovered as independent lineages from the other Asian species of the genus Scolopendra with 100% support and a sufficiently long basal distance. Both morphological observation and molecular phylogenetic analysis consistently show that the undetermined Scolopendra is a new species. The Scolopendra archeona, new species, has approximately 20 centimeters in length and 2 centimeters in width. It is the largest centipede to be found in Japan and Taiwan. And its deep bluish green body is beautiful and remarkable. On June 29, 2001, after the 3 months after the description of the new species, the Ministry of the Environment announced that Scolopendra archeona and 2 species of the endemic cockroach will be subject to the species conservation law in Japan. The original purpose of this study, which is contribute to the conservation of Scolopendra archeona and nature of Yambaru, has been So the second topic is the discovery of new cave-dwelling species from the Wyckhuis. The family Nesticidae are the small to medium-sized spiders that inhabit moist, dark areas such as forest floor, vegetated cliffs, caves, and other subterranean environment. These spiders are a major component of arthropod communities, especially in subterranean ecosystems. And Japan is a hotspot for the diversity of family, as it as a home to more than 20% of the named species. Furthermore, most of Japanese species are endemic to Japan, and their distribution is restricted to small area or even single caves. So the Francesco-san recognized and classified the Japanese species of the genera Nesticera and Hoaya, so-called short-legged nesticid spider, using an integrated approach consisting of phylogenetic analysis based on DNA sequence data and species identification analysis, as well as conventional morphological observation, with special attention to the male and female genitalia. And in the current classification of Japanese spiders, one species of Hoaya and two species of Nesticera were recorded as validly named species. However, as a result of our study, the 3 species of Hoaya and 6 species of Nesticera were identified, with 300% increase of the number of Japanese short-legged nesticid. In addition, 5 of them were new species, all discovered from the Wyckhuis. The Nesticera terrestris, Nesticera brevipes, and Nesticera cervicola constitute a monophyletic group, and Nesticera terrestris is distributed in eastern Japan and brevipes in western Japan, with some overlap in distribution in the Kinki region. And Nesticera brevipes is distributed as far as southern Japan, and its sister clade Nesticera cervicola is distributed only on Yakushima Island. And for Hoaya mogera, 2 major interspecific lineages are distributed north, east, and southwest, separated by Tokara Strait. And Nesticera okinawensis distributed in the central Wyckhuis. The Nesticera ocula distributed on Ishigaki-jima Island, and Nesticera insulana distributed on Yonaguni-jima Island diverge deeply phylogenetically from each other. Therefore, the distributional pattern corresponding to the geographical structure is observed. Of the 5 new species, Nesticera oculata, Hoaya alba, and Hoaya subterranea, are found in the deepest and darkest part of caves and show extreme morphological adaptation to life in the permanent darkness of underground environment, including loss of eye and color. In the future, it may be possible to discuss the evolutionary process of phylogenetic pattern and cave adaptation in the geographical timeline of the formation of the Wyckhuis by conducting more detailed analysis with more gene markers. So even in Wyckhuis, where biota have long been studied, the species diversity of invertebrates living in the forest floor and underground is still poorly understood. In the past 3 years, we have discovered many new putative species and are currently preparing several taxonomic papers. However, unfortunately, our study is unlikely to keep up with the speed of habitat destruction and the extinction of species and local population in the Wyckhuis, as well as other biodiversity hotspots in East and Southeast Asia. Especially the loss of species diversity in caves and underground environment can go unnoticed and required immediate investigation and reasonable conservation planning. So in order to drastically increase the speed up of elucidation of the species diversity in Wyckhuis and as well as other biodiversity hotspots in Asia, and to promote biodiversity conservation, it is necessary to build the enough infrastructure and capacity of taxonomic research. The Okinawa National Museum of Natural History, which Okinawa Prefecture is currently doing its best to recruit, surely become the strong base to support taxonomic research in this area. So country borders have no meaning for organisms other than humans. So organisms that inhabit the Ryukyu have undergone unique evolution through the settlement of ancestral population from the continent, followed by a process of repeated geographical isolation and dispersion. So in order to elucidate the species diversity that lay in the background, it is necessary to form friendly and mutually beneficial academic relationship across multiple countries and regions, and to foster the next generation of researchers under these networks. So Okinawa National Museum of Natural History must serve this function as well. So thank you very much for your attention.
Thank you, Dr. Katsuyuki Eguchi. Please stay online for later discussion. For the next speaker, I will invite Dr. Kris Wyckhuis. I'm sorry for the pronunciation. He is from Chrysalis Consulting, research interest in biodiversity-friendly agriculture. His expert and skill is agroecology, biological control, and insect biodiversity conservation. His P.Sc from Kent University, his master from Kent University, and his doctoral from Purdue University, United States of America. He published 136 international peer review. Dr. Kris, please, the time is yours. Sorry, Dr. Kris is not online. So I will invite Dr. Srinivas. Is Dr. Srinivas online? Okay.
Yes, Pa, I'm here.
Okay, that's good. Dr. Srinivas, sorry, I cannot read from here. Okay, he is from PT Bayer Indonesia. Areas of interest is crop management, resistant management, native resistant breeding, integrated management solution, technical advocacy and outreach, and regulatory advocacy, interest predictive and digital agriculture and small farmholder initiative. His education, Ph.D. from Department of Entomology, University of Nebraska, United States, and major in entomology, minor in biochemistry. His master in agriculture, Department of Agriculture Entomology, Andhra Pradesh, University of Hyderabad, India, major in entomology, and his P.Sc in agriculture, Andhra Pradesh Agriculture University, Faperta, India. Dr. Srinivas Parimi, the time is yours.
Thank you so much, sir. So I get more time of 2 speakers? No, I'm joking. Don't take it.
You still have 15 minutes, yeah.
Yes,
You are
yes.
behind.
Thank you so much. And good afternoon to everybody who is in the conference room there. Firstly, my thanks to Padang for inviting me to be a speaker here, and also the organizing committee for facilitating this in a virtual mode when many of us could not really make it there. Thank you so much. Allow me to share my slides here. And let me see. Okay. Let me know when you can see my slides.
Yes, we can see your slide.
Thank you so much. Thank you so much, sir. So today I'll be talking about insect resistance management, insecticide resistance management, but in short, it's IRM, and how do we do it in general. We know very well that over a period of time, the number of cases of resistance evolution to pesticides has increased. It's never plateaued or it never flattened that curve. So from the database, when you look at it, there are around 17,000 odd cases of resistance reported, out of which, interestingly, 12,500 close to are field evolved and field practical resistance, if I may call it, but field evolved mostly. So that tells you how important is this area of work of around resistance management and understanding resistance evolution in pests. But what is it exactly that we all understand? A very standard definition, but there are few things that we need to understand. Anything that you call as insecticide resistance should be heritable in nature, meaning it goes over generations. And also it should translate into failure of a product, not just one season, but repeated failure. And this product failure is reflective of actually the expected level of control or lack of expected level of control. So in short, if you're calling something insecticide resistance, you need to be able to observe this over time and in terms of product failure for control. How do insects develop resistance to insecticides? To be fair to this entire resistance management and resistance development in insects, resistance is a natural phenomenon. It's nothing that happens out of the blue. And when you look at it, one of these, or at least, let me get the laser here. So one individual out of say 1,000 or 10,000 be having a propensity, or in technical terms, we call that resistant allele present to a certain pesticide. What we do as humans, as growers, is we start spraying insecticides to control some of these insect species. Here, let us assume it is, say, Asian cornborer, rice BPH, whichever one you want to. And so over a period of time, these reds, they start increasing. Why? Because the way we actually spray the insecticide matters. And many times in smallholder systems, and even at times in large farming systems, the use of insecticides is not done as per recommended doses, or it is not done as per the method that you really want them to. But either way, when you start spraying the same insecticide over years and years, season to season, with no change in mode of action, finally you will end up with a situation where you have these all reds. And when you cross this stage 3 is when you start seeing changes happening in the field. Your product doesn't work anymore. So overall, the repeated spraying causes it, but also at times overusing within a season. So there's a habit in the farmers that they generally go for calendar-based spray every 7 days, every 5 days, every 8 days. That is not good. We know that. We talk about it. How much the farmer is doing it is a big question. Added to all these issues is the fake and counterfeit products, which we have in the market today, which we are trying our best to mitigate this risk, but it's not easy without your support as academicians or as public institutes. Most importantly, spraying something at less than recommended dose. This is the most critical one. This triggers lots of phenomena in terms of resistance management. What it does is basically it selects the insect at sublethal doses. And that is very risky in terms of not only to the current product that is being used, but also the future products. So several reasons for resistance development in insects. How do you manage it? And we all know these recommendations, rotate it with different modes of action. Try to not mix products with the same MOA. And of course, products applied in each of these 30-day windows should be different. Lots of suggestions, but what are feasible at the farmer level is the thing that we need to understand. So the most feasible one is to teach the farmer of reading mode of action labeling that is on the labels in Indonesia now. It's a mandatory thing, which is fantastic. So you read it and teach the farmer that, okay, you cannot use same numbered pesticides in the field. Beyond this, as an example, I wanted to show this. When we started developing in Iraq, I was part of this working group in Iraq International. Iraq is Insecticide Resistance Action Committee. It is very interesting to note that we started with IPM. We were supposed to develop IRM guidelines. We started with IPM. So pretty much we went back to our conventional way of managing things, which is very, very, very welcoming. And it is also important that we keep this in our thoughts that IPM today has more prominence, more relevance than any time in our entire careers. So, agronomic actions. If you start with removal of cover crops, volunteers, so which harbor this fall armyworm resting stages, and synchronous planting with first rains, good crop management, removal of the crop residue, and destroy infested plants. All of this contribute positively to fall armyworm management. I don't know how many people here work in corn. Weed management really contributes. Fall armyworm really hides in the weeds, lays eggs there, comes back, feeds on corn, at times goes back and hides in the weeds as well. A good weed management really contributes to good fall armyworm management. This is all IPM, right? So what is IRM component in this? It is again rotation of modes of action and nothing much. Of course, we do have these window-based applications that is gaining prominence in the past few years. I think it's a very good approach to ensure that we have farmers understanding this window-based approach to manage insecticide resistance in crops. Now, I already mentioned about IPM gaining prominence and more so relevance today, and IRM being there using IPM techniques. So they go hand in hand. We believe in Bayer that IPM and IRM go hand in hand. They save a lot of money for us. They save a lot of time for us. It also gives a safer, cleaner produce, and also it protects our health pretty much. So we believe in this, and we are promoting this more and more. And we're also working on many technologies that would actually contribute to the basic principles of IPM. Okay, just shifting gears a little bit, that's related to insecticides. There are BT crops that we're all aware of. Of course, Indonesia does not have a BT crop yet, although it has a HT crop recently released, herbicide-tolerant crop. BT crops are actually considered as extreme cases of host plant resistance. We are all entomologists here today. We know the importance of host plant resistance in pest management. It's super critical. It lays the foundation of pest management with other techniques like cultural control. So the value that BT crops bring into overall pest management is immense. And I'm going to talk about it in a different way later on at the end of the presentation. But keeping this in mind, I want to bring your attention to the BT corn technologies that we have in Asia. Why I'm bringing this up, corn as a crop is grown everywhere in Asia. Indonesia has the third largest area of corn in terms of production as well as corn acreages in Asia. It's a very important crop for us in Indonesia. And there are many technologies in Asia as on date, particularly grown in Philippines and Vietnam and received approvals in Pakistan. Most of the products that are sold in Philippines and Vietnam today are either, VT Double Pro from Bayer, BT11 based AgriShoot project products from Syngenta. I heard there is one more that Syngenta has launched recently, and of course this is from Corteva. So the most sold single gene based products are to some extent this monoitin based product, a very small amount in Philippines, and of course dominated by Syngenta products. So we have a matrix of products that are in Asian countries that are sold as of now, particularly Philippines and Vietnam. We are also coming up with new technologies, which is not the subject of discussion today. But what I want to talk is how do we look at these products in terms of resistance management? A basic diagram. When you plot the increasing dose of Bt protein against increasing mortality, this is how it looks. All the susceptibles are killed, all the resistant survivors are here, and there is a genotype called SR or RS. This is called heterozygote. The expression level in the plant should be so high when you're designing that product that it kills both these genotypes of the insects, whereas this is the only genotype that will be left. Why? Because this is naturally found, and this cannot be really killed even with a very, very, very high dose many times. So you're only aiming to kill these all the time. That is called effective dose, or many times it is called high dose. At times it's called high kill. Whatever it is, it kills both these, genotypes. In this context, I want you to introduce you to one of the classical works or publications from Rick Roche in 1998. He used deterministic modeling with certain assumptions, but I only want to call out three important assumptions. His assumptions, very important one, are resistance is stable and dominant, and the fitness costs are not there for resistance, meaning resistance, if it is found, it is going to be existing in the environment with no fitness costs. That's the first assumption he made. The refuge will provide all the susceptibles, and the single gene or the BT genes will provide enough kill, or at least it kills most of the susceptibles and the heterozygotes. Now, based on these assumptions, let us look at this picture, which talks about resistance evolution as a measure of the frequency of resistant allele reaching 50% or more. So basically, as you go up, it means the frequency increases and the resistance evolution is happening. Okay, and this is the mortality percentage of these heterozygotes that I am talking about, the RS genotypes. The first scenario, if the 95% of the heterozygotes are killed, you will see that the durability of that particular single gene product, this is specifically for single gene product, will be up to 40 generations. This is deterministic model. This is not actual data. Okay. Now this is very, very pleasing to hear. Only if the single gene product can kill 95% above, which we consider as high dose. But how many of those are in the market? We need to be looking at. What happens if the mortality is less than 90%? What is the compromise we would be making? In the first instance here, a 10% refuge can provide or delay the resistance for 40 generations from the time of introduction.
Dr. Srinivas, your
yes,
time, 3 more minutes.
Yes, thank you. Whereas when you look at this, with a 20% or more refuge, you will not be able to protect it beyond 20 generations. When you look at anything less than 50% mortality, no refuge will help. So very critical that you have a refuge planted. Similarly, another model which shows that the pyramided product is better than a sequential release of 2 individual genes. Pyramided products have 2 genes at least with 2 different modes of action, whereas sequential use means if you release these 2 modes of action, products one after another into the environment. In pyramided product, you combine those 2 and make one product. It's interesting to see that Refuge is still necessary for pyramids. Pyramids can delay resistance longer at 10% than a sequential release of products, which would be a single gene released after a single gene product, even with 20 to 40% refuge. So pyramids have a super advantage in that. Same way, even with 30% kill, why I'm saying 30% kill here? I'm comparing this line C, which actually means in the pyramid will provide only 50% of control with one toxin or one gene, and 30% mortality of the heterozygotes with the second one, which is very less. But still, in a pyramided product, it survives, it fares much better than a sequential release. So overall, what it tells you is, if the single gene products do not have high dose kill, there is a huge disadvantage, even if there is a refuge. Whereas a pyramided product, even if it has less than high dose or lesser efficacy, there will be a second gene to kind of help in killing these individuals. So it's called redundant kill. Overall, what it tells you is both these products need a refuge, but they also raise the question of movement of larvae between BT and non-BT plants in pyramided products, particularly in a refuge mode of seed blends, which I'll be talking later. And basically, when a larvae feeds on BT and moves to non-BT, they will survive. So it's a sublethal exposure and survival. But pyramids generally do not allow survival of these larvae on the plants. What if it moves from non-BT to BT? Well, you're looking at large-size larvae that would be feeding on BT plants, but pyramids can take care of it to most part. So eventually these things die. The only risk to this movement is the effective size of refuge will be reduced. Finally, to kind of conclude on this particular topic of which one is better, another modeling study then also says that there is a huge advantage of releasing a pyramided product into a country than a single gene-based product immediately if you look at a target pest. But the most notable thing that you need to be looking at is the cross resistance between these two proteins in a two-gene product. Quickly on the refuge, I want to mention here that most people prefer structured refuge, but our experience clearly tells us in smallholder systems, and for that matter, even in the large farming systems of US and Brazil kind of countries, this kind of triggered a lot of refuge non-compliance. And that kind of escalated the risks of resistance development in target pests. So we in Asia, most of our products, and I think it is agreed among industry now that we go with seed mix or seed blends as a refuge deployment model when you have 2 or more genes, because they can control the pest very effectively, and refuge requirements can be lowered from say 20, 30, 40% kind of refuge requirements to 10. In Philippines, we have 5, of course. And it's also based on the risks that are perceived based on the target pests in the region. So seed blends ensure 100% compliance and also reduces complexity at the farm level. Functionally, on the topic of cross resistance, functionally effective pyramids, of course, delay onset of resistance. For example, Mon89 that Bayer Crop Science has 2 modes of actions, which is like 2 different genes acting at 2 different sites on the target pest. So this is a functional pyramid. Whereas if you take any other product which may have same class of Trivan class or Cry2 class, then it's not a functional pyramid unless it is binding to 2 different sites. So this can hasten the development of resistance at times. So the cross-resistance risks are already proven, for example, in corn against ACB and Falernum. It's an established fact now. So to reduce the risk, we should be looking at technologies. We should be looking at a good, robust strategy that could actually ensure long life of some of these products, and which comes with the refuge deployment. So I have these few key takeaways. Mode of action, rotation is important. IPM, lot of relevance today, not just for pesticides, but also for BT crops. Refuge is a very critical component, and pyramids reduce refuge requirements, but they still need the refuge. And maybe the size of the refuge may be a little smaller or lesser than single gene products. Please note that cross resistance is a huge risk when you look at corn systems in Asia and probably in future for Indonesia. So I'll stop here. Thank you so much. Terima kasih. Look forward to questions, sir.
Thank you, Pak Srinivas. Apparently, we have a video from Dr. Kris, so we'll play the video.
Okay. Yes, sir. I'll mute myself. Thank you.
Okay, thank you.
To join you virtually today for the 11th Congress of the Indonesian Entomological. I'm very grateful to Huda for the opportunity to present today, and I also wish to apologize that I cannot be with you in person. So in today's presentation, I will try to pitch biological control as a low carbon solution. As we all know, anthropogenic global warming has really reached the proportions of a planetary emergency. Yeah? The global food system in particular is a large contributor to global warming, representing approximately 30% of global greenhouse gas emissions, which are equal to 4.9 gigatons of carbon equivalent. Food systems emissions are rapidly rising, especially in developing countries. On-farm production, be it agriculture, aquaculture, or fishing, is responsible for nearly 40% of emissions. And livestock production in particular is a well-recognized contributor of methane emissions. Cropland emissions themselves are primarily ascribed to methane from rice cultivation, CO2, nitrous oxide, and methane from peatland draining, and nitrous oxide from nitrogen fertilizer application. Now, if we aim to decarbonize the global food system, change from the current status quo is really needed. A number of solutions are on the table, which include increase in yield, efficiency, or a drastic reduction in food waste, and a transition to plant-based diets. The latter option entails moderating our intake of dairy, meat, and eggs, and is a powerful solution that can bring about 50% of greenhouse gas reduction. However, what we often forget is that growing plants itself continues to be very energy intensive. For example, in global wheat production, the use of synthetic fertilizers and pesticides is responsible for between 15 and 50% of greenhouse gas emissions. While the carbon footprint of synthetic fertilizers has been very well characterized, pesticides are a blind spot in carbon accounting. Their carbon footprint is entirely unquantified, and they are also under-recognized as drivers of environmental change and biodiversity loss. This, by itself, represents a huge missed opportunity for the promotion of non-chemical alternatives, such as biological control. We try to fill this critical knowledge gap through a relatively straightforward exercise. So how did we go about in doing this? First, we extracted data on the volumes of different types of pesticides that are used across the globe and averaged these values over 2016 to 2018. Next, we computed the carbon equivalents for pesticide production, transportation, storage, and transfer, and we added in energy expenditures for pesticide formulation, field spraying, and equipment manufacturing, while also accounting for seed treatment. Now, based on the data that we were able to access, Recognizing that some countries do not report or underreport, we find that the synthesis, formulation, distribution, and field level use of pesticides generates carbon emissions worth almost 20 million tons of carbon equivalent, with a range between 6.4 and 37.2 million tons. This is equivalent to the emissions of almost 16 million passenger vehicles driven per year, 170 million barrels of oil, or about 18 coal-fired power stations. Among the world's major economies, pesticide-intensive nations such as the United States, China, and Brazil really stand out. Now, let's have a closer look at the pesticide-related carbon footprint in Asian countries. As you can see, of course, China really stands out here, but the carbon footprint in that country is equivalent to that of 1.7 million passenger vehicles driven per year, which is really substantial. Other countries also have large carbon footprints. These are Australia, Malaysia, Russia, Japan, and South Korea. Meanwhile, countries such as Indonesia or Vietnam appear to have relatively small carbon footprints. However, we cannot be fooled because recent research from US scientists shows that these countries consistently underreport pesticide use in global databases. In fact, Indonesia is honestly not doing a good job in reliably reporting pesticide data. The real data for Indonesia are shown here and are obtained through FAO stat data, through older FAO stat databases. And they are honestly quite concerning. As you can see, since the end of the Suharto era in 1998, pesticide use in Indonesia has grown 50-fold. So the carbon footprint is much larger than official data let us believe. This really accentuates the urgency to promote biological control as a low carbon alternative for pesticide for chemical pesticides, and to reduce the greenhouse gas emissions of agriculture. So what does biological control really entail? Biological control is desirable, or a so-called best bed crop protection solution that entails working with instead of against nature. It involves the use of living organisms for the non-chemical prevention or suppression of biotic stressors, be it weeds, pests, or diseases in agricultural or natural ecosystems. There are different biological control modes. One mode involves the conservation or the active protection of beneficial organisms that occur within a cropping system, while a second mode involves the periodic release of laboratory-reared or laboratory-grown arthropods or microorganisms. Ideally, biological control measures are complemented with action at different temporal or at different spatial scales. This could involve the establishment of wildflower strips at grass barriers, or hedgerows at the farm level, or the use of microbial seed coatings or agroecological practices such as the addition of organic matter or manure to the soil. Overall, biological control has a low or even a negative carbon footprint, especially when infrastructures such as hedgerows or cover and intercrops are established, because those directly contribute to carbon sequestration. Biological control organisms thus constitute formidable allies for individual farmers or homeowners, and there are countless vertebrate, invertebrate, or microbial organisms that are ready for use or wait to be deployed. However, their potential is routinely discounted due to an over-reliance on chemical pesticides. Biological control constitutes a sound value proposition, and this is evident, or this was evident under the UN Coordinated Farmers Field School program, which was a decade-long campaign that covered all of Asia and was subsequently replicated in Latin America. The Farmer Field School program involved hands-on training of farmers through innovative observation-based learning approaches and covered several millions of hectares across Asia. Through these campaigns, which centered on the promotion of biological control, one was able to reduce pesticide use by 80 or even 90% in a wide variety of crops. This while sustaining, or as evident in this table, even raising crop yields. Now, when accounting for current production areas of particular crops in specific countries, one can calculate the total emission reduction that could be achieved through biological control. For example, the greenhouse gas emission reduction achieved through effective use of biological control in India's cotton crop can amount to 250,000 tons of carbon equivalent, which is really substantial. Similar values are obtained for the rice crop in Thailand or in Vietnam. Now, if we want to transition away from these energy-intensive pesticides and move towards low-carbon alternatives, one wonders whether the necessary scientific basis is available. In this exercise, we screened the global literature. Specifically, we logged the number of historic or all-time scientific publications for 153 countries on 3 agroecology subfields, being agroecology itself, IPM, integrated pest management, and biological control. Next, we plotted a country's scientific outputs against affluence or the country's GDP per capita. What do we see? The wealthier a country is, the longer its techno-scientific trajectory in biological control or agroecology or IPM. Now, a second question is whether this scientific progress of a given country relates to the country's pesticide use levels. Here we link a country's literature output in the field of biological control to total pesticide usage and to pesticide usage intensity per farmer or per hectare. And what do we see as pesticide absolute and relative usage increases? There is substantial progress in biological control science. Hence, pesticide-intensive nations are best prepared to move towards low-carbon alternatives such as biological control. In principle, biological control science can deliver real-world social ecological outcomes. It can contribute to climate change mitigation by shrinking the carbon footprint of agriculture. It can add to climate change adaptation by increasing ecological resilience. It can benefit biodiversity, and it can deliver a broad suite of desirable One Health outcomes. However, sound science is not enough. So we show this in this exercise, in which we did a reality check. And we looked at the concrete evidence on whether increasing scientific output really translate into actual pesticide usage reduction. And unfortunately, the evidence is mixed, as you can see in these plots, where we relate year by year scientific output to trend lines in pesticide usage intensity. Unfortunately, only 30% of the world's major economies effectively curbs pesticide use. Even scientific powerhouses such as Germany, Spain, Canada, or Australia face progressively increasing pesticide use. So every year pesticide usage increases in those countries. Meanwhile, countries with far shorter scientific trajectories in biological control or in agroecology, such as Colombia or Egypt, these countries do bend the curve. They exhibit a so-called agroecology technique effect, and they engage in leapfrogging. They make smart use of other countries' scientific progress in biological control and implement it in their country to reduce pesticide usage intensity. In those countries that fail to bend the curve, common adoption hurdles are weak policies, sometimes political change, ineffective stakeholder engagement, and industry meddling. And especially the latter, we cannot disregard. Now, if we wish to translate science into practice, there are a couple of rules of thumb, a couple of elements that we can bear in mind. We need to better communicate science. We need to mend public perceptions. We need to shift agri-food production priorities, and we need to tighten regulations. And overall, we have to overcome quite a number of challenges. We need to refute discourse that pesticide-free farming is ineffective or unprofitable. This is quite evident in the work from the Farmers Field School program. We need to abandon monodisciplinary approaches and easy single-factor remedies, such as seed coating, for example. We need to decouple pesticide sales from farmer advisories, and we need to pursue policy change involving both hard taxes or regulatory caps and soft incentive schemes measures. And on policy change, Indonesia honestly can learn from itself. Indonesia really has all it takes to successfully implement biological control. In fact, during the 1980s and 1990s, Indonesia was a front runner, if not the poster child of the Farmers Field School program, and its campaign received strong backing from the president's office. In fact, in 1986, Suharto banned nearly all pesticides in rice and launched an ambitious national policy and a comprehensive farmer training program, which became a model for many countries across the globe. By doing so, Indonesia was able to almost entirely phase out pesticide use in rice production while increasing farm revenue by 19% and almost halving the environmental footprint of agriculture. So in conclusion, what can we say? Chemical pesticides account for up to 5.8% of global cropland emissions, and these products thus directly contribute to global warming. These impacts are further compounded by negative effects on human health, on food safety, on ecological resilience, and on biodiversity loss. As an alternative, biological control constitutes or provides a solid value proposition. It can decarbonize food systems, it can raise farm profitability, and it can favor integrated rural development. Pesticide-intensive nations, including Indonesia, possess credible techno-scientific capabilities in biological control. However, lenient environmental regulation and efficient policies are major hurdles. In order to effectively take biological control forward as a low-carbon solution, sound science needs to be coupled with enabling policies, as demonstrated in 1990s Indonesia. So that's it for today's presentation. If you have any questions, please feel free to approach me by email. I'd be very happy to address your questions. I wish you a good remainder of the conference and a good dinner tonight as well. Hopefully see you soon.
Thank you, Dr. Kris. Ladies and gentlemen, we are 30 minutes behind the schedule. So I only want to take one or two burning questions. Budami is not available for discussion because in United States, it's very early morning. And she said that very cold there, so it's very sleepy. So only one burning question, or one or two burning questions. So please. Okay, so Ibu, please.
Thank you very much. Only a short question for the third presenter regarding pest resistance to insecticide. My question, do insect predators and parasitoid develop resistance to insecticides? Thank you.
This is for Dr. Srinivas, right? Okay,
Yes.
Dr. Srinivas.
Yeah, can you hear me?
Yes.
Okay, that's a very good question. In fact, there were attempts actually to design parasitoids with insecticide-resistant genes. But unfortunately, many of the parasitoids and predators, when exposed to many chemistries, they are hit by the chemistry very badly. So, to answer your question, yes and no. And today's chemistries, which are coming with more, how do I say, greener chemistries, you call them, they are much compatible with parasitoids and predators. But the older chemistries, they may not be so compatible. So, but as a management perspective for pesticide resistance, I think we call them supplemental releases of biological control agents, and we should be doing that if we can. I know it doesn't work at farmer level. Availability of these agents, etc., is a challenge. But again, yeah, to your question, yes and no, based on what chemistry you're looking at.
Okay, thank you. Is there one more questions?
Can I ask one question?
Oh, there's online Pak Intan. There's an online question. Pak Intan, so please.
Okay, thank you, Rama. Dr. Srinivas, thanks for your presentation. I enjoyed very much. Actually, my question related to pesticide resistance, you show the data that's from time to time, it keep increasing, right? And then I know that to develop a new insecticide is very, very expensive. Some people say about 10 million USD up to more than 100 million USD, right? So what would be your, advice how to prolong the use of good insecticide? Otherwise, I know it's also a matter of time, right? I mean, before this resistance happening, but we have to be able to, somehow to make it slower, before it develop resistance. Thank you.
Thank you, sir. Yes, thank you for that question. And the answer will be very big, but what I'll do is I'll call out two important things. There are lots of steps and actions you can take to ensure pesticide durability in the markets. I'm talking about the markets because that's where the entire our technicality applies. I see there is a lot of communication to the farmers missing. How they use those pesticides is missing. From my college days, 30 years ago, it's the same problem. I think that's one of the critical components that will extend the life of some of these pesticides. And I believe in. I have seen, I have worked, I am responsible for Asia Pacific. I have seen many countries, and that's on the farmer side. Now, on the technical side, I think we are doing our bit as industry. In fact, I should appreciate Indonesia, its regulations and regulators who made a mode of action labeling mandatory. It is a very big step for that matter. What it does is it allows us to talk to farmers in certain language, which is technical in a way, and which is easy for him to understand as well. I am hoping this can bring some big change, and once we start pushing this to the ground level. But otherwise, sir, there are lots of suggestions and techniques one could talk about. Pesticide mixtures is one thing. In bringing in biological control is one thing. Pheromone-based technologies, lots of stuff. So IPM integration is one step. I sincerely believe it's at the farmers level and in their hands, and we should ensure that they understand. I think we are missing it now.
Okay, thank you, Dr. Srinivas.
Thank you, Dr.
Last
Srinivas.
question is from a lady in front of stage. I think someone in here. Okay.
Thank you for the time. My name is Meliana Gusti from Biology Department of Universitas Negeri Padang. Thanks to your presentation for Eguchi Sensei. I interesting about insect in Sumatra. So my question is, taxonomy of most the animal group is still poorly understood. If I want to research a genus and that has minimal information, as a researcher, how I should do to start that research? That's my question. Thank you.
Is it for who?
Eguchi Sensei, Katsuyuki Eguchi Sensei.
Oh, Mr. Katsuyuki Eguchi. Okay, Dr. Katsuyuki Eguchi.
So now can you hear me?
Yes.
Okay. Okay. Yeah, a bit difficult question. So yeah, maybe what is the starting point and most important point to start the taxonomic research? It may be her question. So yeah, actually, so there are a lot of the animal groups or organisms of which the taxonomic knowledge is still limited. And so, yeah, actually we need more taxonomic capacity. I mean the infrastructure, and also we need more young generation who are interested in the taxonomic discoveries. And because for example, my case, I study on the ant for more than 25 years, but I just described the around 80 new species and 2 genera. So the discovery by the only single taxonomist is still very small, but maybe now there are more than 200 2 million named species of organisms, and then the real species number is maybe 10 times or 20 times more. So to do the speed up the elucidation of species diversity, maybe we need some advanced technique to detect the putative new species, and also we have a very strong network among the researchers who study on who are interested in the taxonomic discovery. So yeah, I really happy to know that you want to join the taxonomy. So thank you.
Okay, thank you, Dr. Katsuyuki. Yeah, as we know that Indonesia is one of the mega diversity, and we believe that more than probably 50% of insects, especially, probably undescribed in Indonesia. So yes, I think the field of taxonomy is still open widely in Indonesia. Okay, ladies and gentlemen, thank you for Professor Damayanti, Dr. Susan, Dr. Katsuyuki Eguchi, Dr. Kris, and Dr. Srinivas. So let us give a big applause for the presenter. Thank you very much, and I'm sorry for the delay. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you so much for the keynote speakers, and also the moderator, please to take the position, and we will see the moment to give the token of appreciation is also the certificate for the moderators and also the invited speakers. For the invited speakers, please to keep in the Zoom and we will show the certificate. Firstly, we invite the President of Entomological Society of Indonesia, Professor Dr. Dadang M.Sc, to take the place and give the certificate for our moderator today in the plenary to Dr. Purnama Hidayat from IPB University Indonesia. Please, Professor Dadang, and also Dr. Purnama Hidayat to take the position, and we see the moment, the incredible moment of us in the plenary too, ladies and gentlemen. All right, let's give an applause for our moderator, Dr. Purnama Hidayat from IPB University Indonesia. Thank you so much. And please, Professor Dadang and also Dr. Purnama always stay there, and we will see the certificate for our invited speakers that in the Zoom. Firstly, we give the certificate appreciation. Big applause for Professor Damayanti Buchori from IPB University Indonesia. Let's give an applause, ladies and gentlemen. Thank you so much, Professor. And next for Dr. Susan Bay F. Kalumpang from University of the Philippines, Los Banos. Thank you so much. Next is Dr. Katsuyuki Eguchi from Tokyo Metropolitan University, Japan. Terima kasih and arigatou gozaimasu. Dr. Kris Wyckhuis from University of Quensland, Australia. And the last one, the certificate, big appreciation for our invited speakers, Dr. Srinivas, the delegation from PT Bayer in the plenary too. Let's give an applause for all the invited speakers in the plenary too and in international conference. Thank you so much, Professor Dadang and Dr. Purnama Hidayat. Please to take your beginning position. Ladies and gentlemen, we move to the next agenda. Is the photography contest announcement. We giving applause and please welcome Dr. Yusuf Hidayat to announce who is the winner of the photography contest now. Please give applause for Dr. Yusuf Hidayat.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, now we are going to announce the winners of insect photography. There are four categories. Two categories for smartphone and two categories for digital camera. The committee already prepared the video, so please just play the video to see some idea about the photos sent by the participants. Baik, kita lanjutkan dengan agenda pengumuman. Siapa pemenang untuk setiap kategori. Ini adalah Dewan Juri yang telah bekerja keras untuk menilai. Ada 4 kriteria yang dinilai. Yang pertama adalah ini untuk Dewan Juri. The first is Dr. Iputu Sudiarta from Universitas Udayana. Kemudian Insinyur Nur Andriono MT dari UTAB Photography, and Oki Purnama S, AMD Pariwisata, Makro Photographer. Terima kasih untuk Dewan Juri yang telah bekerja keras untuk menilai siapa pemenang dari Lomba Fotografi Serangga PI untuk tahun 2023. Yang pertama, kategori smartphone pelajar. Siapa pemenangnya? Untuk juara ketiga, ini adalah juaranya. Abdurrahman Algoji Ahta dari Madrasah Mu'alimin Muhammadiyah Yogyakarta dengan karya berjudul Hama pada Pohon Pisang. Selamat untuk Abdurrahman atas capaian ini. Abdurrahman berhak untuk mendapatkan hadiah berupa uang sebesar Rp500.000 beserta sertifikat. Berikutnya, untuk pemenang kedua. Pemenang kedua adalah Raufa Hafizul Wafi dari SDN 28 Ampenan Mataram, Nusa Tenggara Barat. Judul karyanya adalah Predator Serangga. Baik, ini yang ditunggu-tunggu siapa untuk pemenang pertamanya. Judul karyanya Setangkai Berdua. Juaranya adalah Ewaldo Abrar Napi Setiawan dari Pondok Pesantren Sabilul Ilmi Subang, West Java, Jawa Barat. Untuk pemenang kedua tadi mendapatkan hadiah uang 1 juta, kemudian untuk juara 1, 1,5 juta beserta dengan sertifikat. Untuk pemenang nanti akan dikontak oleh panitia terkait dengan nanti hadiah dan juga sertifikat. Demikian untuk kategori yang pertama. Oh ya, ada tambahan untuk trofi ya, ada trofi yang nanti akan dikirim oleh panitia. Baik, kategori berikutnya smartphone untuk kategori mahasiswa dan umum. Ini untuk juara ketiga. Karyanya berjudul Si Cantik Koksi Sang Predator. Siapa dia? Arsi dari Universitas Sriwijaya. Berhak untuk mendapatkan posisi sebagai juara ketiga. Berhak untuk mendapatkan uang 500.000, sertifikat dan juga trofi. Berikutnya, juara kedua. Judul karyanya adalah Ngengat Kolibri Menghisap Nektar. Siapa dia? Imam Subail dari SDN. Maaf, ini tempat bekerjanya. Jadi bekerja di SDN 28 Ampenan Mataram, tapi beliau adalah Bapak Guru ya. Selamat untuk Pak Guru, Pak Imam atas karyanya. Beliau juga kalau tidak salah sempat juara di periode sebelumnya ya. Selamat untuk Pak Imam Subail, pemenang kedua untuk kategori smartphone mahasiswa dan umum. Siapa juara pertamanya? Who is the first winner? Serangga Sadis judul karyanya. Siapa dia? Pemilik karya ini juara 1, Dahlan Yuda Pratama dari Bandar Lampung. Selamat untuk Saudara Dahlan Yuda Pratama atas capaian ini. Demikian untuk kategori yang kedua. Kategori yang ketiga, ini kamera digital. Ya. Kamera digital untuk pelajar Siapa dia untuk pemenang ketiga terlebih dahulu? Nah, ini karyanya. Jangkrik sedang berjemur. Ya, jangkrik sedang berjemur. Siapa dia pemilik karya ini? Inilah Almaira Azaria Putri dari SDN Tulung Rejo 03 Jawa Timur. Selamat untuk Almaira, berhak untuk mendapatkan uang 500.000, sertifikat dan juga trofi. Berikutnya juara kedua. Who is the second winner for digital camera? Leptosianina, judul karyanya Muhammad Afrija. Pemilik karya berjudul Leptosianina dan berhak untuk mendapatkan gelar juara kedua dengan hadiah 1 juta, sertifikat dan juga trofi. Juara satunya siapa? Untuk kategori ini Skipper Sang Polinator, itu judul karyanya. Siapa dia pemilik karya ini? Risti Sundari Nusifera dari SMP Tunas Unggul Bandung. Selamat untuk Risti, telah menjadi juara pertama untuk kategori kamera digital pelajar. Mendapatkan uang sebesar 1.500.000, kemudian sertifikat dan juga trofi. Terakhir, kategori terakhir, ekspektasinya dengan kualitas tertinggi. Jadi kategori kamera digital untuk mahasiswa dan umum. Siapa dia untuk juara ketiga? Ini judul karyanya, Pemangsa Hama Belalang, karya dari, this is the third winner, Nanang Suherman from Bandung as the third winner for this category, kamera digital untuk mahasiswa dan umum. Berhak untuk mendapatkan uang 500.000, sertifikat dan juga trofi. Juara kedua. Ini judul karyanya, Kelahiran Sang Penyerbuk. Pemilik karyanya adalah Sudarmanto Edris dari Bandung. Berhak untuk mendapatkan gelar juara kedua. Berhak untuk mendapatkan uang sebesar 1 juta, sertifikat dan juga trofi. Terakhir, juara ke-1. Cantiknya penyerbuk jarak jauh. Juara 1 adalah Made Chandra Aruna Putra dari IPB Universiti. Ada yang hadir di sini mungkin siapa? Made Chandra ada? Tidak ada? Baik, demikian pengumuman untuk kategori keempat kategori. Saya coba cek mungkin apakah di Zoom ada yang hadir pemenang? Pak Subail mungkin, silakan. Silakan, bagaimana Pak Subail kesan dan pesannya, sempat juara di sebelumnya dan sekarang masih tetap masuk 3 besar. Silakan. Pak Guru, Pak Subail.
Mohon maaf, jadi saya tidak bisa menyampaikan ini ya, pesan atau kesan dikarenakan berbenturan dengan kegiatan diklat pengangkatan sebagai kepala sekolah. Nah, dalam kegiatan kali ini, alhamdulillah, saya mendapatkan penghargaan luar biasa ini lagi dari P Pusat bersama ICC. Ini luar biasa. Bukan saya saja selaku kepala sekolah di SDN 28 Ampenan yang mendapatkannya, tetapi juga siswa saya yang tak lain juga merupakan putra saya sendiri yang masih duduk di bangku kelas 2 SD. Itu juga mendapatkan di kategori pelajar. Nah, untuk makro ini bagi saya ini adalah untuk melihat keajaiban, berbagai keajaiban yang ada di dunia yang kecil. Kita melihat secara langsung seperti binatang gajah, binatang apa lagi. Tetapi kalau dunia makro ini kita tidak bisa melihatnya, sulit melihatnya dengan kemampuan, tanpa kemampuan bantuan seperti LITA atau fotografi makro ini. Itu yang luar biasa. Sehingga ketika kita mengaplikasikannya ini menjadi luar biasa. Nah, hal inilah yang jadi dasar saya, salah satunya karena saya juga seorang pendidik. Jadi saya sangat perlu mengaplikasikan makro ini dikarenakan makro ini karena bahan-bahan bacaan yang ada di SD, kebetulan saya saat itu mengajar di SD dan sekarang sebagai kepala sekolah SD, itu bahan-bahan ajar itu selalu tentang serangga-serangga, tentang capung, kemudian kecoa, lebah, kemudian ya nyamuk itu. Sehingga jarang sekali disampaikan tentang serangga-serangga yang lebih unik. Sebagai contoh di kategori smartphone tadi, itu luar biasa keunikannya, juara 1 itu. Milik siapa tadi? Mas Huda ya. Itu luar biasa menurut saya. Itu tidak pernah ada saya temukan di buku. Begitu juga yang saya kirim, yaitu mengat kolibri. Tak pernah saya lihat di buku pelajaran SD. Dari sanalah saya mengonsep pembelajaran di SD itu dengan mengaitkan dengan dunia makro. Bahkan saya tidak ragu untuk mengajak siswa saya untuk berpetualang di lapangan, untuk menjelajahi dunia makro di dalam pembelajaran. Nah, terkait dengan bagaimana proses saya mengambil foto ini kesan saya luar biasa di sini, Pak. Jadi, mengat kolibri, sebelumnya mohon maaf, terdengar ya suara saya?
Terdengar.
Ya, terima kasih. Mengat kolibri ini adalah serangga yang boleh dibilang tidak pernah ditemukan oleh pemakro lainnya di Lombok. Saya pun itu hanya beberapa kali saya temukan.
Ya. Baik, Pak Subayil. Terima kasih atas kesan dan pesannya. Mudah-mudahan tetap bersemangat untuk berkarya, juga para peserta didiknya ya untuk tetap berkarya, termasuk untuk ini ya di bidang insek fotografi. Demikian untuk pengumuman lomba fotografi serangga. Mohon maaf jika ada kekurangan. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. And once again, let's give applause for Bapak Dr. Yusuf Hidayat. And also once again, congratulations for all the winners of photography competition in this event international conference. Ladies and gentlemen, this is the end of our agenda today. We would like to say thank you very much for all distinguished guest speakers and all participants. Also, thank you so much for all invited speakers in this international conference. Also, the sponsorship and partnership in this international conference. We hope you enjoy and please to join this event. Tonight, we have the gala dinner with the governor of Wyckhuis Sumatra. Please be prepared and join us here in this ballroom at the 7 a.m. So, ladies and gentlemen, see you in the second day of International Conference of Entomological Society of Indonesia 2023. See you. Wassalamualaikum warahmatullahi wabarakatuh. And ladies and gentlemen, please to take the position, we have to photo session. All of participants today that join us here in this first day of International Conference. Bapak Ibu yang kami hormati, kami mengundang seluruh peserta, partisipan, invited speakers, keynote speakers, dan semua presentator yang akan presentasi besok untuk dapat berfoto bersama di depan stage. Please to join us to take the photo session in front of the stage in the first day of this international conference. Ladies and gentlemen, once again, we invite all of the participants here in the first day to take the photo sessions in front of the stage. Dan kami mengundang Bapak Ibu tamu undangan yang terhormat untuk dapat berfoto bersama di depan panggung untuk mengabadikan momen luar biasa. This is the incredible moment for us because in this international conference and the 11th Congress of Entomological Society of Indonesia 2023, we have a memorable moment here, and please to take the photo session, yes, in front of the stage. Please, ladies and gentlemen, Bapak Ibu, ladies, Miss, Ma'am, Sir, Mr., all of you, lecturer, the researcher, to join us here in the first day and take the photo session with all the participants in delegation. We have one line in front of the stage and on the stage, please to take and make one line again in on the stage. Bapak Ibu silakan untuk membuat satu barisan di depan panggung dan satu barisan di atas panggung sehingga kita bisa mendapatkan space dan Bapak Ibu semua bisa terlihat di dalam kamera. So let's rejoin for the stations now. Please make a line in front of the stage and on the stage, please. All right, ladies and gentlemen, please keeping smile in front of camera. We count in 3, 2, 1. Again, big smile. 3, 2, 1. Time's up, ladies and gentlemen, in 3, 2, 1. Okay, let's make a video. If I said Ichesi, you guys said the best and go. Ladies and gentlemen, I will count in 3, 2, 1. If I said Ichesi, you said the best and go. All right. Ichesi 2023. Giving applause for us. Thank you so much, ladies and gentlemen. And we have that information for you to join the gala dinner tonight. And also for the oral presentation and the participants can give the content to the link in the group WhatsApp. You can send that your paper or presentations for tomorrow to the link of the group WhatsApp. And also for ladies and gentlemen that has been already to buy a hat and also the outfit, please to take in the Entomological Society of Indonesia booth in outside of this ballroom. Ladies and gentlemen, Bapak Ibu yang kami hormati, bagi Bapak Ibu peserta oral presentation untuk dapat mengirimkan bahan konten presentasi paper ke link yang ada pada grup WhatsApp dan juga bagi Bapak Ibu yang sudah memesan baju serta topi dapat mengambil di booth yang berada di depan, tepatnya di booth dari Entomological Society of Indonesia atau PI. Ladies and gentlemen, thank you for your time and see you tomorrow.